So it always gets a little scary as the list of things we have to do gets shorter. That means we're getting closer and closer to the exam, and that is indeed the case here. We're on still through the cardiovascular system, but we are on to the blood vessels here. Well, we'll talk about their microscopic anatomy and function today, and then we may do some anatomy for that as well, depending on how things are doing on time. We still have a handful of assignments due before the next exam. On Wednesday, your Unit 18 review is due, your cardiovascular exercise is due, and remember that is going to be graded for correctness, and your Physio-X exercise 5, and as I saw earlier, everybody loves those Physio-X's. They are totally awesome and a tremendous fun. I know some people like to do them multiple times just because they're so enjoyable, and this one has 7 activities, so please feel free to enjoy that. On Monday, when we come back from the weekend, you have your Unit 19 review due, your Physio-X exercise 11, only activity 4 for that one, and your hematology labster. Remember, 100% correct and complete and 80% correct for that. Also remember, this weekend is your last big weekend to study for the exam. We are having the exam on Wednesday next week, lab and lecture exam. So this is your big weekend to study. I will try to limit how much new information we have on Monday, but there is going to be some new information provided on Monday. So again, remember though, this shouldn't be new information for you. You have my lecture slides that tell you exactly the things that we're going to be talking about. So there's no reason you shouldn't be looking ahead. Don't let Monday's lecture be completely new information to you that you haven't thought about at all 2 days before the exam. Both exams are timed. You may take them in either order that you want, although like I said before, most of the problems that people have tend to occur during the lab exams. So I would encourage you to yes, if there's time for a review, we should hopefully have time for review. Like I said, I'm trying to front load the information as much as possible so we can do a review on Monday. But remember, Monday, a review is not me telling you the things that I think are important. That's what I do every day in lecture. Review is your opportunity to ask me questions, and then I will help you try to come up with the answers to those. And remember also, if we have a review and you don't ask questions, then I assume you've mastered the material and I make the exams harder. All right, so take them in any order that you want. They are timed exams. So like, for instance, the lecture exam may be 1.5 hours, the lab exam may be 2.5 hours, whatever it is. I haven't finished the exams yet, so I don't know. But remember, they must be completed during the class time. So even if you have 2.5 hours for the lecture exam, if you start the lecture exam at noon, you're only getting 35 minutes. These must be completed during the class time, so please be aware of that. We then have a whole week off. Monday is a holiday, so when we come back on Wednesday the 17th, there's going to be 2 pre-labs that I'm going to be assigning to you that you're going to be doing. But the other very important thing we will be doing on Monday is we'll be forming our groups. Hopefully at this period of time, as we are nearing the end of our first section of 431, you have learned that it is vitally important to be in class, to be active in the learning process. But it is going to be especially important to be here on Monday the 15th, because on Monday the 15th, you are going to be forming your groups and you are going to be picking your topics for those 8 to 12 minute oral presentations that are worth 50 points towards your final grade in this class. And if you are not here, you do not get to be part of a group. And if you're not part of a group, you get a 0 for those 50 points. So it is important to be here and to be present so that we can form those groups, we can pick those topics, and you can be successful on that assignment. All right, questions, and I'll remind you more about that and actually tell you which pre-labs are due as we get closer to that. But I want to plant the seed now so that you are aware of it. All right, any other questions? All right, excellent. Let's go ahead and get started then. Our goal today is to start talking about the rest of the cardiovascular system. We've done the cardio, time for the vascular. We are going to talk about the blood vessels. Now, as we talked about at the very beginning when we identified the components of the cardiovascular system. We mentioned that there are 3 types of blood vessels, but in reality, there are actually 5 types of blood vessels. Ones we know already are, of course, the arteries. And how did we decide we were going to define arteries? All right, my microphone is on this morning, right?
Blood flows away from the heart.
There you go. Blood flows away from the heart. Remember, most arteries are oxygen rich, but most is not all, right? Remember that pulmonary circulation screws it up for everybody. So we do not use oxygen carrying content to define our arteries. We define them by the direction they carry blood, and arteries carry blood away from the heart. Excellent. However, there are also a smaller class of blood vessels that carry blood away from the heart, known as arterioles. Obviously, they are similar to arteries. They are small arteries, but there are some other anatomical differences for them as well. These arterioles are also what are known as the resistance vessels. And why do you think that might be the case?
Are they less elastic?
Great question. It isn't so much that they are less elastic. They are a little bit less elastic. They do have a little bit more muscle to them, but really their job is to modify the resistance of blood flow, right? Blood flow and resistance are 2 opposing forces, and the resistance of the blood vessel doesn't just affect blood flow, but it also affects blood pressure, right? As many of you know, because I've mentioned it either in this class or definitely in 430, I have 2 daughters. My youngest daughter has an extreme peanut and tree nut allergy. And one of the concerns with that is that if she were to come in contact with a peanut protein, her body could have an allergic reaction to that, where many of her arterioles would all dilate at the same time. And if all of those arterioles dilated at the same time, what would happen to her blood pressure?
Decrease rapidly?
Yeah, it would crash, right? Basically, the 2 concerns that you have is that the blood pressure is going to drop, having trouble for her getting blood to her brain. And the other thing that that protein can do is cause the mucous membrane of her respiratory passageway to increase and produce more mucus. So it constricts her airways so she can't get air. Her blood pressure drops dramatically. And what's the fancy word we have for that type of response?
Anaphylaxis.
Anaphylaxis, anaphylactic shock, absolutely, right? So that ability of these tiny little blood vessels to change their diameter has a tremendous potential impact on our blood pressure and our ability to flow blood through our circulatory system. Because after all, what makes the world go round?
Pressure.
Pressure, absolutely. And so that ability to control the diameter of those blood vessels has a dramatic effect on our blood pressure. Now, there's one other way we can define arterioles as well. Arterioles feed blood into our capillaries. Right? I think we talked about this already. All of our arteries, all of our veins are just conduits. What did we say we meant when we used that term conduit? Anyone recall what I mean when I say conduit? I'm pretty sure we talked about this. It's like the PVC pipe carrying the water around your lawn in the backyard.
The media of something going through another thing.
Exactly, like absolutely. Basically, it's just a hallway. It's a passageway. It doesn't make anything. It doesn't modify anything. It is just a passageway, right, to get from one location to another, right? The capillaries are the only place where exchange takes place. And when we talk about exchanging materials, obviously we are talking about exchange between the blood and either the air that is taking place in the lungs, not sure why I'm on cap lock, or in our interstitial fluid. What type of things are being exchanged at the capillary?
Gases,
This is excellent.
nutrients.
Oxygen.
Yep, one of those
Hormones,
gases. What else? Hormones, I heard hormones.
chemical signals. Sorry.
Other chemicals. Any other gases besides oxygen?
Carbon dioxide.
Let's not forget fluids, water, more specifically, not just fluids, but water, absolutely. Metabolites.
And then heat.
There you go, heat as well, absolutely. Although heat is something that is able to radiate out through the blood vessels themselves as well, not just the capillaries, right? So that isn't necessarily something that is exchanged, although obviously as the fluid moves out in the capillaries, then so does the heat move as well. So we can put that one in parentheses. Excellent. So those are all examples of things that are exchanged at the capillaries. Excellent, excellent, excellent. Once that exchange is taking place in the capillaries, capillaries feed into tiny veins known as venules. And those tiny venules feed into veins. And of course, how did we define veins?
Flows to the heart.
Yeah, there you go. Carries blood to the heart. Excellent. Perfect, perfect, perfect. Now, obviously, one of the big differences between arteries and arterioles is size. One of the big differences between venules and veins is size. However, in veins and venules, really size is the only difference. Whereas arterioles are both smaller and anatomically different. So these are basically small veins, whereas arterioles are small, but also different anatomically. There really isn't a big difference. I'm not sure why everything's so huge. There we go. There really isn't a big difference in the anatomy of a vein versus a venule. Really, it's just the overall size of them. But there are some differences between arteries and arterioles, so we will see that. If you were to take all the blood vessels out of your body and stretch them end to end, it would hurt a lot. All right, questions on that? All right, excellent. Let's talk about the microscopic anatomy of our arteries and veins and capillaries. Here we see a pretty picture that shows the relationship of these things to each other. Now, when talking about blood vessel anatomy, and we should specify here, we are talking about arteries and veins at this point. There are three layers, or what we would call three tunics. And as we go from deep to superficial, the first one is what is known as the Tunica Antema. And actually, let's go ahead and draw these because we like our anatomy. Oops, hold on. There we go. Keep hitting the wrong button. I haven't had my caffeine this morning. All right, excellent. So let's go from, let's cheat. Here is our blood vessel. We will start in our lumen and we'll work our way up. As I mentioned, the first layer is what is known as the Tunica Antema or the Tunica Interna. Either of those are acceptable terms. Our goal here is to allow for the smooth, continuous movement of the blood that is going to be in this lumen. We obviously don't have to worry about protection or anything like that. So what type of epithelial tissue do you think would comprise a nice, smooth, simple surface for that blood to flow?
Simple squamous?
Yeah, it's going to be a simple squamous, absolutely. Epithelial tissue. Of course, we're probably going to need a connective tissue for it to sit on top of. And what type of epithelial tissue would be good to hold that simple squamous epithelial tissue in place?
Areolar.
Areolar, exactly. Perfect. Now, this simple squamous epithelial tissue lines the inner surface of our blood vessel. And so what do you think we call this simple squamous epithelial tissue based on its location?
Mesothelium.
Mesothelium, remember, was on the outside of the heart. But remember, we had a smooth, simple squamous epithelial tissue on the inside of the heart, and we'd probably want to continue that out into the blood vessels as well. And absolutely, we call this the endothelium. Excellent. Endothelium within, and then we'll put our squiggly areolar connective tissue on top of that. There we go. Our next layer is the Tunica Media. And what would make a good chewy nougat center for a 3-layered structure? What do we need this blood
Smooth
vessel?
muscle?
Yeah, exactly. Smooth muscle. Excellent. We need a smooth muscle layer. So we're going to have a big chunk, well, not big, but we're going to have a chunk of smooth muscle sitting in here. And what will the function of this smooth muscle be?
Dilate.
Well, I like that. So change the diameter of the blood vessel. Absolutely. One way is by relaxing, in which case it would cause the blood vessel to dilate. But of course, if we were to constrict it, then that would cause a decrease in the size of the lumen as well. Excellent. So we're going to be able to change the diameter, either decrease or increase the size of the lumen. I like that. That's a good way of describing it as well. From that, as we know, because we talked about it, this is controlled how? What controls the smooth muscle of our blood vessels?
Sympathetic nervous system?
There we go. Remember, one of the things we talked about is most organs in our body get dual innervation from both the sympathetic and parasympathetic, but of course, most isn't all. And so in this case, we have the, excuse me, sympathetic nervous system controlling the diameter of most blood vessels. Our third and final layer is the Tunica Externa, or what is also known as the Tunica Adventata. This is going to be a fibrous connective tissue. And why might we want a big layer of fibrous connective tissue sitting on the outer surface of our blood vessel?
Protection.
Yeah, absolutely. One reason would absolutely be protection. We want to provide some integrity to this blood vessel. Why might another good reason to be as well?
To hold the pressure.
Okay, absolutely. I would say that falls into protection, but absolutely, I have to say structure and protection definitely are in there. Well, let's ask the question this way. How many people here have either given blood or taken blood? No one. You're all a bunch of selfish bastards. Okay, excellent. There you go. Someone said they've given blood. Excellent. All right, just once. Anybody given it more than once?
About 2 or 3, 2 gallons, I think.
Oh, wow. That's impressive. Absolutely. Right? There you go. 10 times somebody else. Excellent, excellent, excellent. So for those of you who have given it, do they have to chase your blood vessels around inside of your arm every time they're trying to get it? Or if you're taking it, do you have to chase the blood vessels around inside their arm while they're moving all over the place?
No.
No. The other thing this fibrous connective tissue does is it attaches it to the surrounding tissue. So it anchors it in place as well. Right? So it provides protection and it provides that attachment, holding it in place. Now, This is true for both arteries and veins. Now, are both arteries and veins going to have the same size lumen, the same size muscular layer, and all that type of stuff? No, but there's another big difference between arteries and veins as well. Our arteries only, and what color? Let's use, we'll use orange for this. Yikes, Ash, that's does not sound like a lot of fun. All right. With our arteries only, there are two additional sublayers that are more prominent in the arteries. The first, not toe, two. The first is what is known as the external elastic membrane. And this one is located in the Tunica Antema. So what there is this additional layer of elastic tissue within the Tunica Antema. And there is also the internal elastic membrane. And this is located in the Tunica Media. So within the Tunica Media, we again have this thick layer of elastic membranes. Now the question becomes why? Why might we have all these additional elastic fibers found inside of these two sublayers? It's inside of these two layers. What's the point of having all this extra elastic fibers in an artery? Right. So excellent. Not only do we want to allow stretch, but what's the other side of elasticity?
Recoil.
There you go. Excellent. To allow stretch and recoil. The reason for this is our arteries are the higher pressure blood vessels. They're the ones that directly receive the blood from the heart. And as we just learned from talking about the cardiac cycle, is the heart like a faucet that you turn on and it flows continuously? No, it pumps blood in a pulsatile nature, where it forces blood out of the ventricles, and then it stops, and it forces blood out of the ventricles, and it stops. When it does that, as we saw, we see dramatic changes in the pressure of the blood. Right? And you haven't thought of it in those ways, but if you wanted to measure someone's heart rate, would you have to hook them up to an ECG so that you could watch the movement of the heart, the electrical activity of the heart, and see and count the heart rate that way? Or what else could you do if you wanted to feel know somebody's heart rate? Just listen to their chest.
Pulse points.
Yeah, exactly. Pulse points. Right, where you can put your fingers on pulse points, and as you put your fingers on the pulse points, you can feel the pulsatile nature of the blood vessels as they are expanding as blood is forced into them, and then they are recoiling as a result of that. The ability of the arteries to be able to stretch and then recoil allow for a more continuous blood pressure, right? If the blood vessels were very hard and rigid, when the blood was forced into there and the blood vessels had no give, the arteries had no give, your blood pressure would spike dramatically as a result of that. Of course, conversely, as soon as the heart stopped forcing blood in there, the blood pressure would drop dramatically. And those types of dramatic changes in blood pressure would not be good for the movement of that blood. Instead, what can happen is as blood is forced into it, they can stretch to reduce the dramatic increase in pressure. And then as they recoil, they can help to maintain a more continuous pressure. So that ability to stretch and recoil helps to maintain a more continuous pressure. Yes, there are differences in pressure when the blood pumps and when out of the heart and when it doesn't, but it reduces the extremes of those and keeps a more continuous blood flow going. All right, let's go back to the pretty pictures from your textbook and see this. So again, we have that Tunica Antema, the internal layer. And again, there's, oh, sorry, in the internal layer, we have the internal elastic membranes. I thought I had that backwards. I was busy talking and I wasn't thinking. So I apologize. That makes way more sense. That the internal layer would have the internal elastic membrane. And the outer one would have the external. So sorry about that. Didn't feel right while I was saying it, but I was too busy talking to think about and correct myself. I also spelled media wrong, but you get the idea. Let's look at the pretty pictures and forget my drawing. Excellent. So Tunica Antema, we have the endothelium, which is continuous with the endocardium, that's simple squamous epithelial tissue. A subendothelial layer, which is basically a combination of the basement membrane and areolar connective tissue. And then in our arteries, we have our internal elastic membrane. Next, we have our Tunica Media, which, as we mentioned, is smooth muscle, and that smooth muscle is controlled mostly by the sympathetic nervous system. Do that. And in the Tunica Media, we have our external elastic membrane. And again, these elastic membranes help to allow for more stretch, more recoil, to accommodate the more dramatic changes in blood pressure that go along with the heart cardiac cycle. And then lastly, outer layer, the Tunica Externa or Tunica Adventitia, which is a mostly fibrous connective tissue. Now, when we see these two sitting side by side, like in a pretty picture like this, it's usually pretty easy to tell which is which. So as we look at this one here, which is the artery or which is the vein? The one on the left or the one on the right is the vein? I'll ask the question again. Of these two blood vessels in this picture, which is the vein? The one on the left or the one on the right?
Right.
Right. How did you know?
Because it says it.
Because it says it, exactly, absolutely. See how easy that question was? Should have been way more people answering right on that one, absolutely. Now, pretending that it didn't say that there, how else would we know?
A larger lumen.
Absolutely, right? Arteries, as we've already talked about, deal with the higher pressure blood. Veins have much less pressure blood. And remember, as we talked about, pressure is what makes the world go down. Pressure, and more specifically, pressure gradients, the difference in pressure between two environments, is going to determine how easily the blood is going to flow. Since veins are lower pressure, we need a larger lumen. Remember when we were talking about that AV node, the atrioventricular node, we talked about how the cells were very thin, very small, and as a result of them being very thin and very small, they had a very high resistance. So very slow flow. We don't want a slow flow in our veins. We want the blood to be able to move more easily back towards the heart. So typically when you have two blood vessels at the same level, the vein typically has a larger lumen. Notice also, even at the low magnification, we can see that our artery has a much thicker tunica media, much thicker muscular layer to again deal with that higher pressure blood. But notice also, even at a low level, let's use white for this. Notice, actually, let's not use white, let's use green. Notice, even at this low magnification, along the edge, we can see that dark line. Notice at the outer layer of the tunica media, we can see this really black, dark structure. That structure that we're seeing there are the elastic membranes. the internal and the external sublayers of that. So those elastic membranes that make them up. So let's take a closer look at our arteries. Thicker wall, especially the Tunica Media. Then we are going to see in the veins. Much more smooth muscle, much more elastic fibers, and that we see those two layers here inside the Tunica Antema, and then on the outside of our Tunica Externa as well. Another way we can tell at a high magnification view is that when we look at the lumen, because of all of the elastic fibers in our arteries, right, because the blood is forcing them out, when we process this on a slide, there's no blood holding those out. And if you have a rubber band that's being held out and you let go, it recoils. And so that recoil of the elasticity causes the lumen of our arteries to have this ruffled or pleated appearance to its endothelium. Whereas a vein, it will typically be smooth. Now, again, is it bumpy like this in a real body when it's doing its function? No, absolutely not. So remember, this is something we call an artifact, right? Something that is just a result of our processing of this tissue. But it is a consistent artifact and one we can typically use when identifying our arteries. By comparison, when we look at our vein, as we see here, again, larger lumen to it, that's definitely always a good giveaway. Notice we can still see some black fibers inside of here. So it isn't going to have zero elastic membranes or elastic fibers, but it's not going to have near the extensive elastic membranes that the artery has. And again, this is just an illustration, but we can see it when we look at a higher magnification view. That smooth endothelium is usually a dead giveaway that you're looking at a vein as well. Now, you're not going to see this in a cross-section view like this, but also veins have a lot of issues. They're furthest from the heart, so they're dealing with the lowest pressure. They have the smallest pressure changes, so it's really hard to get blood back to the heart. And because we always assume anatomical position, most veins have to carry blood up against gravity back to the heart. So not only do arteries get the advantage of the heart pumping right behind them and the higher pressures and the bigger pressure gradients, but the arteries are carrying most of the blood down. Veins have to carry most of the blood up. And so there are things to assist it. The larger lumen is obviously one of the things that is going to help in that. But the other things that our veins contain are valves. These valves are almost identical to the semilunar valves. They have 3 half-moon face cusps that in their resting position are closed because blood pressure is holding them closed. And when the pressure behind them increases, it forces them open and pushes the blood forward. They look a little like this. So here we see an example of that semilunar valve. Half moon shaped cusps. Again, we only see 2 in this illustration, but there are 3, just like our semilunar valves. And just as our semilunar valves, when the pressure is great enough inside of the vein, it forces that valve open. Notice veins also tend to be more superficial and run along our muscles because we can use those muscles to do work for us. Here is a two-bellied muscle. Anybody remember what this two-bellied muscle is called again? And this is 431. I will take away participation points if you say calf.
Truro?
No. Someone was about to say it. Say it again.
Gastrocnemius.
Gastrocnemius, absolutely. There is the two bellies of our gastrocnemius, absolutely. And as we know, when a muscle contracts, that muscle changes shape. And so notice that's what happens here. When our gastrocnemius is relaxed, right, there's less pressure on that vein. But when we stand up on our tippy toes, and what would that motion be called again?
Pronation?
No, not pronation. There we go. Daniel's got it. Plantar flexion. That plantar flexion of the foot, when we plantar flex the foot, then as a result of that contracting that muscle, the muscle gets shorter, the muscle gets fatter, it squeezes the vein. And as it squeezes the vein and increases the pressure, that forces the blood up into the next part. Of course, when the muscle relaxes and the pressure goes down, the blood wants to go back down into that low pressure environment, but the valve closes and won't let it. So the same way that those semilunar valves in the heart maintain the one-way flow through the heart, here these semilunar valves in the veins help to maintain the one-way flow of that. Now, of course, one of the problems with these valves is they can undergo pressure and extreme pressure. If you're notice we need to contract these muscles to do that. But what if instead you work a job where you're standing for 8 hours inside of a toll booth collecting tolls? Are you getting a lot of opportunities to contract your leg muscles when that is occurring? No, and if the blood congests enough, one of the things that can happen is these valves can weaken. And as these valves weaken, the blood can actually flow back in, causing a large distension of that vein. And what do we call that condition when the valves start to fail and we get that distension of the blood vessels as a result of that? There we go, varicose veins. Those varicose veins are a result of that, absolutely. Excellent. All righty. Here is the pretty picture that kind of shows the anatomical differences between all of our blood vessels. Notice there are two different types of arteries. We'll talk about that in a second. And the arteriole and the veins and the venules. So we see all of those. Again, are these necessarily numbers that you have to memorize? Do you need to know that the diameter of an elastic artery is 1.5 centimeters? Again, I know that kind of information is useful when you're trying to woo women in a bar. You know, if you saddle up to a woman next to a bar and tell, hey, do you know the diameter of an elastic artery is 1.5 centimeters? They just swoon when you say those kind of things. No, these are not numbers that are necessarily important that way. But again, it does show you some of the nice differences. But let's talk about, yes, exactly.
I love that.
I love that. All right, excellent. All right, let's talk about these in a little bit more detail, starting first with arteries. Arteries come in two flavors. The first are the elastic arteries. These are also known as the conducting arteries because they're the ones that are primarily responsible for conducting the blood to different parts of our body. Remember, as we talked about last time, we have somewhere around between 5 and 6 liters of blood in our body. Is that necessarily enough for every cell of our body to all work at their maximal level of activity all at the same time?
No.
No, which is why you shouldn't be, eating Thanksgiving dinner while running a marathon, right? So we want to distribute the blood, conduct the blood to the regions of the body that need it. And our elastic arteries are the ones that do that. These are the ones that are the closest to the heart. And because they're the closest to the heart, they're the ones that have to deal with the greatest pressure changes. We will actually look at the numbers of these pressure changes as we work our way through the blood vessels, and we'll talk about things along those lines. But as we talked about, I think we talked about it last time, blood pressure. We had a systolic and a diastolic. Remember, the systolic was the pressure in the arteries, the elastic arteries, when what was going on?
Contraction.
Yeah, contraction of the heart, absolutely. And the diastolic is when the, well, more specifically, we should be more specific now, when the ventricle is really ejecting blood. So this is, the systolic is the contraction of the ventricle, but really when the ventricle is ejecting the blood. And then the diastolic is when the ventricle is relaxed. And what did we say in normal healthy blood pressure was? Yeah, 120/80, 110/70, something along those lines, right? Absolutely. Notice it's almost a, a 40% increase in pressure from 80 to 120, 50%. It's a 50% increase in pressure from 80 to 120. That's a major change in these pressures. So that ability of these elastic arteries to be able to expand and recoil in response to that, not surprisingly, they have the largest and the thickest layers of the elastic membranes to accommodate those massive changes in pressure that are taking place. So these are our largest arteries and the ones with the most elastic membranes in them, or the largest elastic membranes in them. Elastic arteries then feed into our muscular arteries. Muscular arteries are distributing arteries. They're the ones that, again, distribute it to the different parts of our body. They make up most of the named blood vessels in our body. So most of the blood vessels on your list are muscular arteries. Now, am I going to ask you which ones are muscular and which ones are elastic? No. But what's an obvious elastic artery?
Aorta.
Yeah, aorta, absolutely. Right. And so not surprisingly, as the name indicates, muscular arteries have the thickest tunica media, and they're the ones that give us the most control on the distribution of the blood. If these muscular layers, the tunica medias, constrict, that is going to reduce the lumen and decrease blood flow to that area, whereas when they dilate, they're going to increase blood flow to that area. So they play an important role in distributing the blood to our different parts of our body. Again, perfect example of this is, and again, I have to work on my analogies because we're online, but as I mentioned, in about a month or so, you're going to be giving group presentations. And if you were standing in the front of the class like you're supposed to be doing, giving these group presentations, and 5 minutes into that presentation, your pants fell down, would there be a change in the diameter of the muscular arteries that, say, fed the surface of your skin? Yeah, absolutely, as a result of that. And you'd get very flushed and embarrassed as a result of that. Absolutely. Laura apparently has no problem being naked in front of everybody, but everybody else might be a little embarrassed by that. She's a mom. You have no more. Once you have kids, you just don't have any privacy anymore. I understand. I'm there with you. Exactly. Now, as I mentioned, arterioles are definitely smaller arteries. Again, typically less than 30 microns in diameter. Again, not a number you have to worry about memorizing, just know that they're smarter arteries. However, as we look at the illustration, we can see that our artist has tried to do a good job of showing us the other big difference with arterioles. Arterioles, unlike arteries, do not have 3 complete tunics. Now, as we talked about, arterioles are resistance vessels. These are the ones that can have an impact on our blood pressure. But as you can see, they have an intact endothelium. They have a simple squamous epithelial tissue. But notice they have an incomplete tunica media. Instead of having a complete tunica media, instead, they really have bundles of smooth muscle. Let's say it this way, irregular bundles of smooth muscle around them. So it's not a complete tunica media. And they have scant, which means, again, very little to no tunica externa. So there's almost no tunica externa, or just a scant layer of it on top, and a very irregular tunica media. So not only are they small arteries, but there are actually anatomical differences between them as well. And again, remember we said the other key, that is their anatomy, As for their function, not only are they the resistance vessels, but as we also talked about, their job is to feed blood into the capillaries. All capillaries are fed into by arterioles. Capillaries are our smallest blood vessels, and they comprised of only one tunic. They are basically just the Tunica Antema. Essentially, they're a simple squamous epithelial tissue plus the basement membrane. As we know, all epithelial tissues have a basal lamina or basement membrane that attaches it to the underlying or to the surrounding tissue. Which again, if you think about it, makes sense. This is where the exchange of material takes place. This exchange is passive. Someone remind me again what passive means?
Doesn't require energy.
Yeah, it doesn't require any ATP, any energy for this. And so, of course, as we learned in 430, one of the ways we can facilitate diffusion, we can make that diffusion happen more readily, more passive diffusion happen more readily, is by having a very short distance. So a single flat cell is the shortest distance we can have to separate these two locations to help to facilitate that diffusion of material. Now, again, we have a very simple picture of a capillary here, and it'd be awesome if they all looked like that. But the same way that there are two types of arteries, there are indeed three types of capillaries. So let's take a look at those. The first and most common are what are known as continuous capillaries. Again, notice as we look at the illustration, we can see that as I described, it is a single layer of flat cells, squamous cells, with its basement membrane basically holding it together. Now, notice in this case, while it is a simple squamous epithelial tissue, there are minor spaces in between the epithelial cells. These tiny spaces in between are what are known as the intercellular clefts. And these intercellular clefts are where the solutes, the water, and the stuff dissolved in water are able to sneak into and out of the capillary. Notice one more thing about our capillaries. They're tiny. So tiny that, as you can see here, our red blood cells basically have to line up single file to work their way through them. So obviously those intercellular clefts are not going to allow things like red blood cells or large proteins or other big formed elements to get into and out of the capillary. And that's actually a good thing. Don't we want to limit what can get in and what can get out of our capillaries? Yeah. In fact, in some cases, and notice they've given us an example of it here, we can actually form tight junctions in those intercellular clefts, making it even more restrictive, making it even harder to get things in and out. Where might something like that be useful? Where might we want to be really restrictive about what type of things get in and out of a capillary?
The skin.
Okay, skin, sure, because that's our outside barrier. That could be someplace. Although I was thinking, there we go. I was thinking Daniel's got it. How about in the brain? Remember, we talked about that this is tissue up here that does not regenerate. As a result of that, we want to limit what type of materials get out. So not only do we form really tight junctions on these, but we also take a special type of neuroglial cell to wrap around these and help to form that blood-brain barrier. And anybody remember what particular neuroglial cell was responsible for forming that blood-brain barrier?
Schwann?
Schwann did wrap around, but I think that was the one that formed myelin. What were the ones that formed the blood-brain barrier? They were the most common ones in the central nervous system, had kind of a star-like appearance to them.
Astrocytes.
There we go, astrocytes. Excellent. So absolutely. So we have the ability, there are a lot of these in the skin, a lot of these in muscles, but we can form these tight junctions in them to be more restrictive in places like the brain, helping to form that blood-brain barrier. Again, this is really, really useful because this limits what is able to get into and out. Small things, sodium, ATP, nutrients, things along those lines. But what if we have larger things we want to get into and out of our blood vessel? This wouldn't be helpful for something like that. So instead, we have a second type of capillary, what is known as a fenestrated capillary. Fenestrated capillaries, notice, have a complete intact endothelium with intracellular clefts, with a complete basement membrane. But notice there are holes or pores in the cells themselves. Those holes in the cells themselves are what are known as fenestrations. And these fenestrations will allow larger materials into or out of the capillary. Where might capillaries like this be useful? Where might we want larger things to be able to get in and out? Digestive system, that's a good example. Where else? Someone was about to say something. Were you about to say digestive?
Muscles, maybe?
Not so much muscles in this case. Muscles are more not going to need big, large things. Kidneys are going to be a great example. Endocrine glands, some endocrine glands are going to be examples of places like that. So digestive system, some endocrine glands, the kidneys. What was the choroid plexus again? Anybody remember what the choroid plexus was?
Makes cerebrospinal fluid.
There you go. It makes the cerebrospinal fluid. Absolutely. Right. Where we have fenestrated capillaries surrounded by what type of glial cell? Anyone remember? The epithelial type of glial cell? Ependymal cells. Excellent. Excellent. So basically, ependymal cells wrap around them, helping to make our choroid plexus for that cerebrospinal fluid. But notice, these fenestrated capillaries will allow more things in and out. But notice, again, big things like that red blood cell isn't getting in or out of that capillary. And that's a really, really good thing. Or is it? Might we ever want to get a red blood cell out of our capillary? What do you think? Might we want to get a red blood cell out of our circulation? On occasion, why? Why might we want to do that? There you go, if it's damaged, right? Or if it's sick or dies. Do you all have the same red blood cells in your body that you had when you were a baby?
No.
No. In fact, a red blood cell only lasts about 100 to 110 days. All right. As a result of that, those dead and damaged red blood cells, we absolutely want to get out of circulation. And more importantly, we want to get more red blood cells in to replace them. So for those types of things, we need a highly specialized type of capillary. And this highly modified, highly specialized type of capillary is what is known as a sinusoid capillary. Notice, not only do these have large fenestrations in them, but notice they have an incomplete endothelium. They have an incomplete basement membrane. This has a lot more space, a lot more gaps where very large things are able to get in and out. So let's think about this. If we wanted big things to get in and out, like new blood cells that we're making, right, or get rid of the damaged one, what are some of the organs where we would want this sinusoid capillary?
Kidneys?
Kidneys, well, kidneys, no, we want to filter the blood, but the kidneys aren't where we filter red blood cells. Think of it. Do you have red blood cells that come out of your urine? No, so we don't necessarily want that happening there. There you go, bone marrow. Bone marrow is where we make all of our formed elements for the blood and deposit that in there. Where do we get rid of red blood cells? Anybody know? When a red blood cell is done and broken down, liver is one place, and where else? Spleen, there we go, excellent. So spleen, liver, certain endocrine glands that are going to be producing big hormones, things like this are these areas where we're going to find these sinusoid capillaries. So they're not common, but in the areas that they're located, they're very, very important. All right, lastly, all capillaries feed blood into, I lied first. We're going to talk about how these capillaries are arranged. Capillaries are arranged into structures called capillary beds. Let's draw a really simplistic example of this. We know that arterioles feed blood in, venules feed blood out. So we have an arteriole. That feeds blood into a capillary, and a venule that receives blood from the capillary. And the capillary basically has two components. The first of those components is a structure that is known as the vascular shunt. The vascular shunt basically is a direct connection from the arteriole to the venule. However, off of that vascular shunt are going to be these elaborate interconnected capillaries that are known as the true capillaries. And one of the keys to this true capillary is at the entrance to all these two true capillaries, there is a little circle of smooth muscle. These are known as the pre-capillary sphincters. And while sphincter is an incredibly fun word to say, what is a sphincter? What does it actually do? What is its function?
Opens and closes.
Yeah, it's a valve, exactly, right? Right now, most of you are sitting in a chair or laying in your bed listening to this lecture. Maybe some of you are standing, maybe some of you are on like a treadmill or something like that, but most of you are sitting. And so while you're sitting, right, do your legs need the maximum amount of oxygen and nutrients that they need while you're just sitting there doing nothing? No. And so in that case, we don't necessarily need a lot of blood going to your legs because not a lot of exchange is taking place. So what that would mean is that these pre-capillary sphincters would all be closed and blood would basically just be flowing straight through. However, in a few minutes, we're going to take our break. And when we take our break, like me, you're going to run downstairs to make your coffee so you can run back upstairs. And while you're doing all that running, then the muscles of our legs are going to need more oxygen, are going to need more nutrients, or need to get rid of more wastes. And so at those points, these sphincters can then be opened, dilating to allow more blood flow in and out. So in this fashion, we can control how much blood is going into exchange, how much exchange is taking place, by how much blood we allow into these true capillaries. And of course, how are we going to control these capillaries? How do we control these sphincters? Sympathetic nervous system and
Hormones, chemical signals.
hormones. There you go, hormones. Excellent. And hormones. Perfect. There you go. All right. Here's a slightly more realistic looking arteriole in, venule going out, this big capillary bed. Notice again, we have that thoroughfare channel, which again is another name for the vascular shunt, that thoroughfare channel, and then all of the true capillaries that come off of it. But again, here's a pretty light picture like the one I drew. Arteriole feeding in, venule feeding out. We have that thoroughfare or vascular shunt going down the center and all the true capillaries. And as we talked about, when there is less need for oxygen and nutrients, we can contract those pre-capillary sphincters, decreasing blood flow into the capillary bed. This would decrease the exchange that takes place. Or we could dilate them, increasing perfusion, increasing exchange, typically in response to an increased activity. And again, this is where all the exchange takes place. Then the blood goes back into a venule first. And again, your book doesn't do as great a job of showing this, but venules do have all three tunics to them. So really, they're just small veins, literally just small veins, right? The only difference between a venule, a medium-sized vein, and a large vein is the size. I know they haven't drawn a muscle layer to this one here, but I promise you it is there. All three, venules, medium, and large veins, are just different in size, and I don't care that you know the size differences. Anatomically, they're all the same. As we talked about, all of these tend to have larger lumens. That is going to help to assist in the blood flow. Less resistance, more blood flow. All right. So our blood is constantly being circulated throughout the body, but not evenly. Notice that only about 15% of our blood is in our arteries and arterioles, where 60% is in our veins and venules. Yeah, 12% in our pulmonary, 8% in our heart, only 5% in our capillaries. Our capillaries are the most numerous. I see your question, Ryan. Hold, give me 1 second. We have the most capillaries more than any other blood vessels, but remember, they're so tiny that the blood vessels have to line up single file to get through there. So that's why it's such a small amount. But most of the blood is located in the veins. Go ahead and ask your question now.
Yeah, sorry, I may have missed it, but going back to the continuous capillaries, did you mention anything about the parasites?
What do you mean?
Like what are they and what are their functions?
Did you say, oh, you said pituocytes, didn't you? Or the pinocytes?
Parasite. The
Parasites. Parasites, these are just support cells on the outer surface, like surrounding. So parasites that way. These are cells that may help with attachment. These are cells may help to regulate ion functions, things along those lines. There aren't anything significant. That isn't anything to worry about. And I know it's not shown in the other capillaries, but other capillaries will have them as well. It isn't something we have to worry about. I think they're just trying to point out that there are other cells that are going to help to assist in regulation of ion concentrations and things along those lines. All right. Great question, though. All right. So Notice the huge disparity between blood in our arteries and blood through our veins. Does that mean we have more veins than arteries?
No.
Yes, turns out we do. We do have more veins than arteries, absolutely. Now, it isn't 4 times as many, so that isn't the only reason that makes it up, but we do have more veins and venules than we have arteries and arterioles. But why else is there the big disparity between these two?
Larger lumens.
Part of it is the larger lumens, absolutely. With veins having larger lumens, they're going to be able to hold more blood inside of them, absolutely. So that's the second reason. And there's a third reason as well. Pressures, absolutely. Remember, as we talked about, arteries are dealing with higher pressure blood, and that higher pressure blood moves faster. Veins are dealing with lower pressure blood, and so that blood moves slower. The fact that we're dealing with lower pressure bloods, and again, I'll give you the numbers for this, and these are going to be numbers that are important, and we'll talk about the why that's so significant then. But veins have much lower pressure in them, and so the blood moves more slowly, and that's also why we need more veins, because even minor fluctuations in pressure can impact the movement of blood through a vein. So by having multiple routes for that blood to take back, it's going to help to facilitate the movement of blood there, right? Again, depending on where you live, if you live somewhere in the Sacramento area, there are probably a dozen different paths you can take from your house to the supermarket. So if there's an accident on one street, or if there's, a mudslide or snow or something along those lines, you have alternate routes you can take. If you live up in the mountains in Auburn, right, there may only be one main road that gets you to town. And if something happens on that main road, what are those logging trucks overflow or falls, or crashes or something along those lines, you're not getting to town anytime soon. So having alternate routes allows for more efficient movement of the blood. So they have a larger lumen, we have more of them, and they have lower pressure, so the blood moves more slowly. So while we're at rest, there is a large amount of blood in our veins. However, when that bear with an axe walks into the room, right, one of the things that happens is we can have that venous constriction that we talked about to move the blood back to the heart more rapidly, so we can get it to the muscles and get it to the lungs and get it to the heart, so that we can distribute it more efficiently to deal with that fight or flight situation. So the veins work as a reservoir, but in times of need, we can constrict them to move that blood more efficiently. And again, we don't think of it in those terms, but when someone gets scared, one of the things that happens is they get pale in color. The reason they get pale in color is because all those veins are constricting, moving the blood away from the skin, moving it towards the heart, moving it towards the muscles, moving it towards the lung to deal with that stress response. All right. Questions on that? We have a little bit more we need to talk about for veins, but I think this is a good stopping point. Let's go ahead and take our first break here. We'll come back in 15 minutes, and in 15 minutes, we'll pick up the lecture from here. So any questions before we take our break? All right. I will see you guys in 15 minutes. Go get your coffee, go get something to eat or drink, and I'll see you back here at 9:38. Let's dive back in. Okay, so as we were talking about, the venules definitely have, or the veins really, in all of them, have the tougher road to hoe. They have more of the blood in them, larger lumens, but they have less pressure to help them move it along. So there are ways that we can assist them. One of the ways, as we've already mentioned, is the large lumen. We keep mentioning why this is important, But again, when we talk about this from a physics standpoint, the advantage of the large lumen is that less blood is in contact with the wall of the vessel. It is that contact in the wall with the vessel that causes that friction, and that friction causes resistance. So with a large lumen, we have less resistance. And as we have less resistance, that means we are going to have more flow, easier flow through those. So again, this is all about physics as to why that large lumen is important. Speaking of physics, as we've talked about, there is this relationship between volume and pressure. And those venous valves and muscular pumps help in assisting with that. As we know, when our muscle changes shape, as the muscle changes shape, it squeezes the blood vessel, the vein. I guess we should be more specific. And as it squeezes that vein, obviously volume decreases. And as that volume decreases, the pressure increases. And that increase in pressure helps to force the blood through those one-way valves. Right? Again, notice when we increase the pressure, technically, blood just wants to go away from that pressure increase. However, thanks to the one-way valves, it is not able to go backwards in this case. So it can only move forward, opening that next valve and working its way back towards the heart. But our muscular pump isn't the only type of pump that helps with this. We also have what is known as the respiratory pump. Now, I know we haven't talked about the respiratory system yet, but I know most of you know most of this information. So let's real quickly, oops, I need that, but what I need is my whiteboard. As we know, we have our abdominal pelvic cavity. And as we've talked about, in our abdominal pelvic cavity, there is an organ that acts as a dividing structure in our ventral body cavity. And what is that structure that anatomically divides our ventral body cavity?
Diaphragm.
Diaphragm. And as we know, it is a bell-shaped curved muscle. And of course, when a bell-shaped curved muscle contracts, like all muscles when they contract, it gets shorter. But when a curved muscle gets shorter, what happens?
Flattens.
It flattens out, exactly. So what's going to happen as it contracts is that it is going to flatten out. Now, this is going to help us to breathe because as we've talked about, when it flattens out, then what happens here is the volume in our thoracic cavity increases. And when the volume increases, what happens to the pressure?
Decreases.
Pressure decreases. And because our lungs are connected to the outside world, right? Oops, oh, you get the idea. Then we know that air is going to rush in, right? That's how we breathe in. But notice this also affects the abdominal pelvic cavity. When that diaphragm goes down, notice in here, the volume decreases. And when this volume decreases, our pressure increases. And it's the same thing that happens when the diaphragm relaxes. When the diaphragm relaxes, here in our lungs, our volume decreases, our pressure increases. And of course, as we know, in this case, air is going to move out. But that also leads to pressure changes here. Our volume increases and our pressure decreases. These pressure changes, and not just the thoracic, but the abdominal pelvic cavity, also help in respiration as well. Notice when the pressure decreases in the abdominal pelvic cavity, that is going to help to draw blood up the veins into the abdominal pelvic cavity. Once the pressure increases, that blood's not going to want to stay here anymore. But again, because of the one-way valves, it can't go back to the legs or back to the digestive system. So it is going to help to push the blood towards the heart. These changes in pressure in our abdominal pelvic cavity is what we call our respiratory pump. And it also helps to keep blood flowing back towards the heart, which again, you may not have thought of it in these terms, but you may be aware of it, because if, for instance, you have a rapid and dramatic increase in the size of your abdominal pelvic cavity, because over Christmas break or over the 10 months of quarantine, you've put on 75 pounds, or maybe because you got pregnant. As you get pregnant and that belly swells, that increase in the space taken up by that baby decreases the amount of pressure change that you can produce in your abdominal pelvic cavity, making this respiratory pump less effective. One of the consequences of this is you have a harder time moving the blood back towards your heart. So one of the common problems associated with pregnancy is swelling, especially in the legs and feet, and an increase in varicose veins formation that can occur during pregnancy because of that decrease in the ability to move that abdominal pelvic cavity. So that decrease in breathing and movement that can cause by that is because of that as well. Yeah, absolutely. So it's in the extremities, absolutely, the hands, the feet in particular, and even varicose veins that can form, all because females get the honor and the privilege of carrying babies till full term, growing a basketball inside of them. All right, so this is what we call our respiratory pump. All right, lastly, as we mentioned, we have far more veins than we do have arteries, because we want to have those alternate routes. The more different routes you have to get blood to a location, if there is congestion in one area, then we can send it to an alternate area. And that's really the key here. This is going to help to reduce congestion and maintain a more consistent flow of blood. All right. Let's talk about some of those alternate routes. We, of course, because anatomists love to name everything, the term for these collateral routes to a location is what is known as an anastomosis. IS is, of course, the singular. ES is the plural, anastomoses. There are a bunch of interconnected blood vessels that give an alternate route for blood to flow. And again, this is going to reduce congestion and help to maintain consistent flow. Now, not surprisingly, most anastomoses are located in the veins. Of course, what was the key word in that sentence?
Most.
Most, because most means not all. One example of a location where you might have an arterial anastomosis might be your knee. Why might it be useful to have an anastomosis in your knee? An arterial anastomosis in your knee. Well, let's think about this. If you just had one blood vessel that carried all the blood you needed to your toes, and then you sat down in this chair for 4 hours to listen to my lecture, and you bent your knee while you were sitting there in your chair listening to the lecture for 4.5 hours, what happens when you kink a hose?
You cut off pressure.
Yeah, you stop things from flowing through it anymore. And is that what you want to have happen? When you bend your knee, do you want all oxygen-rich blood no longer going to your toes?
Nah.
No, or your elbows or your shoulder. No matter what position you have your elbow in, you want blood to be able to get to its destination. So these arterial anastomoses provide alternate or collateral pathways to give blood through a given region. Joints are one of the major regions where you see these arterial anastomoses, but the joints aren't where you find my favorite arterial anastomosis. My favorite arterial anastomosis is this one right here, known as the Circle of Willis. Why might this be called the Circle of Willis?
Good old
Bob Willis.
Good old Bob Willis was the first person who described this. Absolutely. Let's take a look at this. There are three main blood vessels that bring, well, really four main blood vessels that bring blood to the brain. The first two, as I'm sure you know, because I'm sure all of you have already started looking at your blood vessel anatomy and learning that blood vessel anatomy as we go through these, are of course the left and right internal carotids. So the left and right internal carotid arteries bring blood to the brain. The other primary blood vessels that bring blood to the vein are the right and left vertebral arteries. Now, because I know you're comfortable and have already started learning this material, what does the internal carotid arteries branch off of? Where do they receive their blood from?
Brachiocephalic artery.
Close. So it will be the brachiocephalic, or for the right side, not necessarily for the left. But what do they immediately receive their blood from? Anybody know? Internal carotids and the external carotids both get their blood from the same blood vessel. What blood vessel feeds both the internal and external carotid arteries? You guys are instilling me in a whole lot of faith on that cardiovascular exercise that's due in 2 days.
The aorta.
Common carotid. There you go. The common carotid artery. Absolutely. Common carotid feeds both the internal and the external carotid. Where might the vertebral arteries, I won't ask you the hard question of where they come off of, but where do they travel? What's distinct about the right and left vertebral artery that you learned way back in 430? Where do they travel to get to the brain?
Inside the vertebrae.
Inside the vertebrae, on those holes on the lateral aspect of the cervical vertebrae. And what was the correct name for that bone feature that they traveled through?
Vertebral foramen.
Vertebral foramen. Excellent, excellent. See, it hasn't all been forgotten. Excellent. Now, there are a couple general paths that the blood takes. For starters, these right and left vertebral arteries come together, and when they come together, they form what is known as the basilar artery. This basilar artery, singular artery, then splits to form the right and the left. And again, this is an inferior view, so this would be the left. Posterior cerebral artery. And guess what the posterior cerebral artery feeds?
Posterior side of your brain.
Yeah, the posterior portion of the cerebrum, absolutely. Excellent. The right and the left internal carotids branch to form two primary blood vessels, one that feeds the front part of the brain and one that feeds the lateral side of the brain. So it basically branches to form, and I'm going to stop writing it on both sides, but I will emphasize it here, the left anterior cerebral artery and the middle cerebral artery. So notice for the most part, we have these direct routes to the different distinct parts of the brain. However, if the blood going to the right side of the front of your brain gets a little sluggish, or the pressure should drop in that a little bit, it's really not that big of a deal if that right frontal lobe is deprived of blood for just a brief period of time or doesn't quite get as much oxygen as it needs, does it? Is it a big deal if your nervous tissue is deprived of oxygen for a brief period of time?
Yes.
Yeah, it's a huge issue. So while these are the primary routes that blood will take, we want to maintain consistent pressure and consistent blood flow to all parts of the brain. To do that, we have three connecting or communicating blood vessels. We have two, I'll use pink for this, posterior communicating arteries. To help to move blood from the front of the brain to the back of the brain, or from the back of the brain to the front of the brain. And we have an anterior communicating artery. Now, again, these are arteries, but they're a little bit special. How many different directions can blood move in this blood vessel?
One.
One, right? Arteries always carry blood away from the heart. So this is going this way, and that's way too big. This is going this way, and 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 this way, and that way, and so on and so forth. However, these communicating arteries can actually carry blood in either direction. So if the pressure is higher on the left than the right, blood can flow to the right to equalize it out. If the pressure is lower in the front than in the back, pressure can move towards the blood can move to the front and back. So in these communicating arteries, blood can go in either direction to make sure that we maintain consistent pressure and consistent blood flow to all parts of the brain. And I have done a truly amazing job of drawing this. However, here is a slightly less accurate, did I do that? Yeah. Artistic representation of what that looks like as well. So again, notice we have the two vertebral that feed into the basilar, feed into the posterior cerebral, our internal carotids going into the middle cerebral and the anterior cerebral. But here we have our anterior communicating artery. Here we have the two posterior communicating artery forming this arterial anastomosis known as the Circle of Willis, which I might point out are all blood vessels you're responsible for on your anatomy list. All right, questions on that? All right, most anastomoses are venous. There are some that are arterial, but guess what? There are even some arterial venous anastomoses, right? And technically, that vascular shunt, that thoroughfare pathway, is an alternate route for blood to get from the arteries to the veins, And so technically that's an example of what is known as an arterial venous anastomosis, where arteries connect to veins. All right, time for some physics. Again, as we've talked about, the goal of cardiovascular system is to move blood. So we need to learn some rules of physics. Blood flow is directly proportional to the difference in blood pressure. Again, let's think about this. I have two blood vessels, blood vessel A and blood vessel B. Assuming they're the same length, assuming they're the same diameter, so everything is the same this way. If the blood pressure here is 110 and the blood pressure here is 50, which blood vessel is going to have the faster movement of blood, top or bottom?
Top.
Top. Top. I've had 3 people say top and 32 people not answer. Now, fourth top. I see some people not answering. Why aren't some people answering?
They're sleeping.
Okay, and that's a very good answer. Some of them definitely could be sleeping. Or maybe some of them are clever enough that they realize they haven't had enough information yet. What if I tell you that on this side, the pressure is 100, and on this side, the pressure is 10? Now, which blood vessel has the higher rate of flow?
Bottom now.
Now it's the bottom, right, exactly. So notice it isn't the absolute pressure that determines flow, it is the difference. It's the exact same thing we talked about with concentration gradients when we were in 430. The higher the concentration gradient, the faster the movement, and it's the same thing here. So it is the difference between the two. And here's the other thing to remember as well. These are inversely proportional to each other. Flow equals 1 over the pressure gradient, delta P. So if the pressure gradient doubled, what would happen to the flow? Oh, sorry, not 1 over, directly proportional. If the pressure gradient doubled, what would happen to flow?
It would double.
It would double. If you cut the pressure gradient by 1/3, what would happen to flow?
Same thing.
Yeah, it would drop by 1/3. So the same way you change the pressure, the same way you affect the flow. All right. Now, of all of our blood vessels, which blood vessel has the most dramatic changes in pressure within them? From one end to the other?
The aorta?
Arteries.
Arteries. Yeah, you're right in the aorta, but the arteries more specifically. Absolutely. And so again, as we talked about, the arteries have the greatest pressure changes, and that is where we have the fastest moving blood. Now, also in our elastic arteries, as we mentioned, we have that pulsatile nature of the blood. And again, you haven't thought of it in these terms, but I know you're aware of these things. Because if you have a really superficial cut in your finger that just cuts a vein, you have a slow, continuous movement of blood. But if it's a deep enough cut that it hits the artery, what happens then?
Blood gushes.
Yeah, but is it gush continuously?
Pulsating.
No, it squirts in that pulsatile nature, absolutely. So it has that pulsatile nature as the pressure changes. We see this in the pretty chart that we have here. Notice a couple things. Notice we have our pressure up here at the top. This highest point pressure, as we've talked about, is what is known as the systolic. And remember, systolic means that this is when we have ventricular ejection. This is when blood is being forced out of the heart into the arteries. And again, that is occurring because our ventricle is contracted. Our diastolic pressure, remember, as we talked about, is when the ventricle is relaxed. And so notice, as we mentioned, in the aorta, in those elastic arteries, we have that good, healthy blood pressure of 110 over 120 over 80. Notice the further you get from the heart, the less pulsatile it becomes through the arteries, through the arterioles, until we get to the capillaries. Now notice one other thing as well. In the arteries, our pressure changes from 120 to about 35. How big of a change is that?
Big change.
Okay, come on. You don't even have to take off your shoes and socks to be able to figure out 120 minus 35. What is that? You're right, it's a big change, but what is it?
85.
65.
Yeah, 85 millimeters of mercury. It's a huge change from aorta to capillaries. Notice in the capillaries, pressure changes from about 35 to about 17. How big of a change is that?
13.
18.
18, absolutely. And then notice, from the time the blood exits the capillaries into the veins, it starts at 17. And by the time it reaches the right atrium of the heart, the pressure is 0. So in all of the artery, all of the veins combined, what is the pressure difference from when it leaves the capillaries to when it reaches the venules? What's 17 minus 0?
17.
17 millimeters of mercury. Notice the smallest gradient is actually in the veins. The arteries have 85 millimeters of mercury of pressure gradient to work with to move blood through it. And the veins have 17 and are fighting gravity to do it. So you can see why we need all those specializations. We need the muscular pump. We need the respiratory pump. We need those valves to help the veins to do their job. All right, it's these changes in pressure from one to another that make the difference. All right, let's talk some more physics. Flow, as we know, is directly proportional to the change in pressure, but flow is also indirectly proportional to resistance. Resistance is the opposition of flow. Right? It's basically a measure of the amount of friction on the blood that slows it down. So in this case, if resistance doubled, what would happen to flow?
It would have.
Yeah, it would have. Conversely, if you were able to drop the resistance to 1/3 of what it would be, what would happen to blood flow?
Increased
Blood flow would triple.
Yeah, it would triple as a result of that. Absolutely. So they're inversely proportional to each other. So the question becomes, what causes resistance in our blood vessels? And there are 3 main factors. Oh, I don't know why those are all coming up at once. Okay, so we'll do it this way. The first, we'll go ahead and put them all on there. What the heck? The first is the viscosity of the blood. What do I mean by that?
Thickness and stickiness?
Yeah, the thickness and stickiness, right? Basically, how much water do you have in it versus how much formed elements? Now, if you're stranded on a deserted island for 3 days with nothing but salt water around you, and you're not drinking a lot of water, is your blood viscosity going to change?
Yes.
If you move up in altitude to the mountains and live there for a couple months, you're going to start to produce more red blood cells to help to maintain the oxygen carrying capacity of your blood, and that's going to change. However, unless you are artificially pumping, right, blood doping by putting a pint of blood directly into your blood vessels, do we get dynamic moment by moment changes in blood viscosity? No.
Happens gradually.
Yeah, it happens much more gradually. It can have long-term implications, but it's not dynamic changes from moment to moment. Same thing with total blood vessel length. Does your total blood vessel length change from moment to moment?
No, it takes time like growth.
Yeah, like times with growth. You get bigger, you get taller, you get fatter. What is one of the things that I think I read somewhere, for every 10 pounds of weight you put on, you add a mile of blood vessels or something. You know, it's one of those crazy, things that they're always throwing out and stuff like that. Yes, as you're changing size, as you're changing weight, as you're growing and doing those things, the number of blood vessels you have will increase and decrease. But again, it isn't something we can do on a moment to moment basis. So the primary way we are going to change the resistance and therefore change the flow of our blood from a moment to moment basis, as Allison pointed out earlier, is the diameter of our blood vessels. This is the one that is going to allow for dynamic, rapid changes in our resistance, which are going to lead to dynamic, rapid changes in our blood flow. This is not, actually, I'm going to put that back. This is not an equation you need to memorize, but resistance is 1 over the radius to the 4th power. So let's think about this for a second. If you doubled the radius of a blood vessel, what would that do to your resistance?
Decrease it.
How much?
I suppose a lot isn't the answer.
Well, yes, but let's actually do the math. 1 over 2 to the 4th power. You all have smartphones in your pocket. Someone figure that out to me. What's 2 to the 4th power?
16.
16. So notice your resistance would decrease by 1/16, meaning your flow would increase 16 times. Now, again, the point of this isn't to make you memorize this, but notice with that huge of a dramatic change, does it take massive changes in diameter to have big effects on blood flow?
No.
It does not.
No. So even minor changes in the blood vessel diameter can have a huge impact on resistance and therefore a huge impact on blood flow as well. And so just even minor changes in the diameter of a blood vessel can have a huge impact on our movement of blood. All right, excellent. Now, obviously, when we talk about the pressure of blood, we're talking about the force of the blood pushing on the walls of the blood pressure. Typically, when we want to measure this, we measure this in one of our systemic arteries, those large elastic arteries that are near the heart, like the brachial, right? Things along those lines, femoral. I guess technically you could do it on the common carotid, but it's really hard to get people to put one of those blood pressure cuffs around your throat, right? When measuring blood pressure, that was a joke, y'all look way too serious. When you measure blood pressure, it's expressed in, and I've already said it, but give me the units.
Millimeters of mercury.
Yeah, millimeters of mercury, how far it can move, a bubble of mercury through space, that millimeters of mercuries. And again, it is those differences in the blood pressure that move blood from one location to another. So blood pressure is important, right? Understanding the blood pressure and knowing the blood pressure is definitely something that is important. We'll talk about how you measure your systolic and diastolic in a moment. I'm sure most of you have had a blood pressure taken, and many of you have maybe even possibly taken a blood pressure yourself. So, as we talked about, we are going to measure, and we'll talk about how we do in a second. The systolic, what was the systolic a measure of again?
The pressure exerted on arterial walls
Right, when?
during systole.
Ventricular contraction.
Okay, and I like all of those things. Obviously, systolic is during systole. Blood pressure is when the ventricle is contracted. But not only is it when the ventricle is contracting, but remember, it's when the ventricle is forcing the blood out. So it's the blood pressure, when the ventricles are ejecting blood out into the arteries. Yes, this is when the ventricle is contracting. But is it the whole time that the ventricle is contracting? Is it the whole time it contracting and ejecting blood? No, most of it. But remember, there's those little bit of time at the beginning when we have that isovolumetric contraction when it isn't. So I'd kind of accept ventricle contracting, but really ejecting the blood is a more accurate measure. And as we talked about, the diastolic is when the ventricle is at rest. Excellent. So if we wanted to get the average or the mean arterial pressure, then we just have to add those two together and divide it by 2, because that's how we get an average, right? Systolic plus diastolic divided by 2, and that gives us our mean arterial pressure.
Don't you have to multiply the diastolic times 2?
Well, so you would be add the systolic and diastolic together, divide them by 2, right? If I wanted to know the average of 2 of your scores, right, and Laura got a 120 and Timothy got an 80, I'd add them together, I'd divide them by 2, I'd get 100, and that would give me the average, right? Isn't that what a mean is? Isn't a mean the average?
Not in this case. It doesn't work like that.
I feel like it doesn't account for something.
Well, but if you look at the chart, isn't that what the chart shows? That the mean pressure is right here at 100 in between. Oh, you're right. It doesn't account for everything. Why not? Why isn't this accurate?
The lumen of the veins are larger.
That's not a bad guess, but let's think of it this way. Instead of taking the average between Laura and Timothy, let's say I was taking the average of Laura, Timothy, and Daniel. Again, I apologize for using your names. You're the 3 that I see on the top on the list here. So again, Laura got 120. Both Timothy and Daniel got 80. So I add 120 to 80. I divide it by 2, and I get 100. And that gives me the average for the class, right? Isn't that the average for the 3 of them? Because in between 80 and 120 is 100. Is that how math works?
No.
No. Notice, yes, 100 is the middle point between 120 and 80, but I have 2 people with scores of 80 and 1 person with a score of 120. Well, let's think of it this way. Do you think that the heart is, oops, not what I wanted to do, is ejecting blood for the exact same amount of time that it is not ejecting blood?
No.
No, it is ejecting blood for a shorter period of time. If they were doing it for equal times, then yes, we would just take the average of the 2 and put them together, and that would give us the average. But that's not what's happening here, because it's contracting, ejecting the blood for less time than it is. So we need a more sophisticated way of calculating the average. And luckily, such a thing exists. All you have to do is measure your cardiac output and multiply that by the total resistance of all the blood vessels of your body. So I'm going to give you guys 3 minutes to do that. In 3 minutes, I want you all to calculate your cardiac output, the total resistance of all the blood vessels of your body, and then tell me all what your mean arterial pressures are. Is that going to be easy for you guys to do?
No.
No. Luckily, there's an easier way to do it. The easier way to do it is to take your diastolic blood pressure and from that, add 1/3 of your pulse pressure. Uh-oh. What the heck is the pulse pressure? We have something else we have to define. What is the pulse pressure?
The difference between the systolic and diastolic pressures.
Perfect. Absolutely. The pulse pressure is the difference between the systolic and the diastolic. Sometimes the pulse pressure is actually a more important value than actually the systolic and diastolic by themselves. So the pulse pressure itself can be an important number. But in this case, what's the difference? Let's do the math on this. What is our pulse pressure for this typical 120 over 80?
40.
40.
40, excellent. So it's 40. What's 1/3 of 40?
Is that 13?
Yeah, let's go with 13. 13 is a good number. So that's 13. What's our diastolic pressure? 80. So notice 80 plus 13 gives us about 93 millimeters of mercury. And notice that is indeed what our mean arterial pressure is. So because the heart is not in a state of systole for the same amount of time that's in a state of diastole, we have to do a slightly funky equation to figure out what our mean arterial pressure is. It's in a state of diastole longer than it's in a state of systole. So we have to do some funky math. Again, we could get the absolute value by taking your cardiac output and your resistance, but while it's easy to figure out your heart rate, how many people know their stroke volume? And measuring the resistance of our blood, all of our blood vessels in our body would be something that'd be really challenging to do. Technically, it would give it to us, but that's a lot of work. So instead, to calculate our mean arterial pressure, which is our average arterial blood pressure, we take our diastolic blood pressure plus 1/3 of our pulse pressure. All right, and that is how you would calculate that. And that is an equation you need to know. Yes.
What did you say because the diastolic or the systolic is in?
The heart is in a state of diastole for longer than it is in a state in systole. So because the two times aren't equal, we can't just add them together and divide by 2. And that's why we have to do this more complicated math. Okay. Awesome. All righty. So we know how to calculate a mean arterial pressure. What we need to do now is figure out how we would calculate the systolic and diastolic. As I said, I'm guessing everybody in this class has at least either had their blood pressure taken or given a blood pressure to somebody else. And of course, what is the name of the instrument that is used for that?
Sphygmomanometer.
Sphygmomanometer.
There you go, sphygmomanometer, absolutely. And remember, spelling does matter in this class. All right, don't say blood pressure cuff. You're not getting any points for that. Sphygmomanometer. And I know that's not how you pronounce it, but that's the way I say it because it helps me remember how to spell it. And because I know Daniel used the term earlier, when using a sphygmomanometer, what is it that you're listening for?
The carotid cough sounds.
There you go. You are evaluating, listening for the carotid cough sounds. That is that little whooshing sound. Now, again, if we were in the classroom, you would actually be taking each other's blood pressures and you would have the opportunity to do this. But somebody explain to me how you take a blood pressure.
You use a stethoscope to listen until you start hearing the the beating sound, and then that's your first
Okay,
number.
but what do you have to do before that? You got the right idea. So we have a blood vessel.
You inflate the cuff first.
Excellent. We have this blood pressure and then a blood vessel. And then basically what we do is we take this big inflatable cuff and we wrap it around that blood vessel and we inflate it. When we inflate it, what does that do?
Stops the
Compresses the
flow.
vessel.
Yeah, basically what happens is it compresses the blood vessel. And when it compresses the blood vessel, blood stops flowing. All right. Again, this is usually when I make the joke that when you guys are working together in groups to do this, you put that cuff on and you inflate it to a good 300 and 350 millimeters per mercury, right? Just to make sure you close
Ouch.
it entirely. No, you don't go that high, but you get about 160, 150, somewhere around those lanes, and it stops the blood flow. Then what do you do?
Slowly release the pressure.
Excellent. Then what happens is you're slowly going to release the pressure.
Eventually, the pressure will be great enough for you to actually be able to hear the sound.
Excellent. Although it's really the other way around. What happens is you slowly release the pressure, and you're absolutely right. Eventually what happens, let's say we get to the point where it drops to 119 millimeters of mercury. As we know, when the heart is ejecting blood, what is the pressure in that blood vessel going to be?
120.
120. Notice when the pressure of the blood is greater than the pressure in the cuff, what's going to happen is that pressure of the blood is going to be able to force some blood into the blood vessel. Now, is it going to move smoothly in that blood vessel?
Now I have turbulence.
No, it's going to produce a tremendous amount of turbulence when it's doing that. And that's that whooshing sound, that Korotikoff sound that you hear as it's going through. Of course, as soon as the pressure drops to the diastolic, drops below that systolic, and we said that that was about 80, then it collapses again. So what happens is we get this short burst, a pulse, let's say it that way, of blood into the artery with lots of turbulence. And we can hear that. And so we hear it as a whooshing sound, and then another whooshing sound, and another whooshing sound, and another whooshing sound. Meanwhile, the pressure is continuing to drop. And as the pressure continues to drop, eventually the pressure drops to, let's say, 79 millimeters of mercury. And what happens at that point?
No more turbulence.
Right. It can know the cuff no longer collapses the blood vessel. And as a result of that, no more turbulence and no more sound. So when you first hear the sound and when you last hear the sound, those are the two pressures you're looking for. The first sound would be your systolic. The last sound would be your diastolic. Your book actually does a really nice job of describing this. So does your lab manual. Like I said, we would actually be doing this activity in the class if we were in the classroom and had the opportunity to do this. So you would have that opportunity. And again, your book's got a pretty picture that shows this process as well. Of course, once you have your systolic and your diastolic pressure, then you can also calculate your pulse pressure as well. And here's the pretty picture from your textbook that kind of shows you that process.
Excuse me,
All right.
Professor.
Yes.
Is there a difference between taking the blood pressure with, let's say, using the arm and that of the legs? Because I've seen some people like take blood pressure within the legs. Does it have like an accurate rate?
So you're a little further from the heart. So typically the blood pressures are a little bit lower. Also, the legs are a little bit larger. So it can be a little bit trickier to hear, like for instance, the femoral artery, because it's deeper, can be a little bit harder to hear those Korotikoff sounds. You have to be a little bit more careful with where you're putting your stethoscope. it works the exact same way. So it is definitely something that's functional. And so if you have somebody who, for instance, has arm injuries or some other reason, then they can take it in the leg. But it's more common in the arm, but there's nothing wrong with doing it in a leg. You would still get a fairly accurate reading. It's just a little bit more complicated to do, a little bit more challenging to do, but otherwise it should work fine. All right. Questions on that? All right. Now, again, this is information that changes almost daily as people have different interpretations and again, nutritionists have different feelings from, phlebotomists and all those types of things. So I'm painting with very broad strokes here when we talk about these types of things. But in general, a normal range is a systolic between 100 and 140. Diastolic is somewhere between 60 and 80, is a fairly broad normal range. And again, this doesn't, this is up for interpretation, but also this is going to vary dramatically by age. And again, I don't want to get that complicated into all of that. We've got plenty of other stuff to worry about, so we're not going to worry about that. Hypotension is typically thought when you have a systolic below 100 millimeters of mercury, but acute hypotension can occur anywhere from as small as a 20 millimeter drop in mercury, right? There you are laying in bed, listening to my lectures, right? With your eyes closed, earbuds on, half asleep as I drone on and on and on. But then I say it's break and you jump out of bed to run downstairs and go get some coffee. And as you jump out of bed, you get a little lightheaded. Is it because of all that vodka you had for breakfast this morning? It could be, but what else could have caused that?
Sudden oxygen demand, and you got a decreased blood pressure, so your body isn't ready for it.
Exactly. While you
Orthostatic.
while you were laying vertically, your heart was not having to beat as hard to get the blood to all parts of your body. So your heart was beating relatively softly. As soon as you stand up, now it's going to take more pressure to force that blood to your brain. And there's going to be a brief delay while the heart compensates. And that delay can cause a slight decrease in blood flow to the brain, as small as 20 millimeters of mercury. And it can be acute, but it can be enough to make you slightly dizzy as a result of that. Right? If you think back to third grade, right? When you were in third grade, it was much, much harder to get alcohol than like fifth grade or sixth grade was. So what would you and your buddies sit around and do, right? You'd all get together in the room, right? With the door locked to keep parents out, curl up into a ball, right? Stay there for 30 seconds and then instantly stand up, right? And as you instantly stand up, you got a little dizzy as a result of that. And it was almost as good as doing alcohol, right? So again, not quite as good, but it was the best you could do at that time. It was third grade after all. Yeah, and in fact, if it drops too much, you can actually pass out as a reminder that is where. Well, I grew up in Ohio, so again, these were the things that we had to do back there. We didn't have fun things like water slides, so instead we had to get drunk. All right. Questions on that? Again, I appreciate these numbers are very broad. Oh, and of course, hypertension is typically, and again, these numbers vary, and we also have pre-hypertension now. Again, I'm painting with broad strokes. Don't come at me with these numbers, but again, these are good broad strokes. Hypertension is typically a systolic above 140 or a diastolic above 90. All right, questions on that? All right, that is everything I wanted to do from a lecture standpoint today. I wanted to finish a little bit early today with lecture because with all the physiology and all the processes we have in this class, unlike 430, we do not have the opportunity to go through all of the anatomy together. However, I do want to spend a little bit of time talking about the blood vessels. I want to talk about some of the basic routes of the blood vessels, but what I really want to focus on are some of the asymmetries. One of the nice things is that left and right, for the most part, are symmetrical. There's a lot of symmetry in our blood vessels. What happens in the right arm is identical what happens in the left arm, but it's not completely symmetrical. There are some asymmetries, and there are also some unique blood pathways that I want to spend some time talking about. So I want to very briefly talk about some of these basic routes and some of these unique routes that we will have. Not having the opportunity to go through every blood vessel on your list. You are responsible for every blood vessel on your list, but I do want to talk a little bit about that. So what we're going to do after the break is come back and spend a little time on these pathways. And hopefully those of you who may be struggling with the cardiovascular exercise, this information might help you to continue to be successful with that. All right, so any questions before we switch gears from lecture to lab? All right, excellent. Let's go ahead and take our next break. We'll take another 15-minute break. So that means come back at 10, what is that, 52? Yeah, that works. 10:52, and at 10:52, we will restart. We'll start lab, and I will start the recording at that point too. All right, any questions? All right, see you guys in 15 minutes. Before we get into the gross anatomy of the blood vessels, I wanted to come back to another nice microscopy slide, again, showing us the differences between the arteries and veins. It can be a little easier to distinguish them when you have them both together in an image. It's even easier when you find an image that's labeled. I just did a quick search and found this one. And even though it was labeled, I was fine using it. But again, we can see both the layers and sublayers of this. In particular, we can clearly see the nice distinct internal elastic membrane. And again, the nice thing about that elastic membrane is it kind of shows the pleated edge of these. Again, it is easy to distinguish them, easier to distinguish them when they're side by side. But if I just showed you a picture of an artery by itself, or just showed you a picture of a vein by itself, you should also be able to distinguish them as well. And again, notice some other things. Pretty much adipose is fine throughout the body, and pretty much also anywhere arteries and veins go, nerves go along with them. So again, we can see those layers, the sublayers of that. And that is the second histology slide you're basically responsible for. So like I said, there's not a ton of histology in this one, not like the next section, which is going to have a lot of it. There's just a little bit, but I guarantee there will be some microscope slides on this exam. But what I want to focus on with the rest of class today, like I said, is the pathways of the blood vessels, our vascular system. So let's bring that lecture up. Again, your blood vessels are listed in your textbook, in your lab manual, in all sorts of other places. Also, I don't have an image of it, but your textbook's got some really excellent flow charts that do a really nice job of showing, instead of schematically, they actually show a nice flow chart of how the blood vessels relate to each other. And remember, that is definitely part of what you need to know. You need to not just be able to identify a blood vessel by a chart or a model or an illustration or something like that, but you also are going to be responsible for being able to know how it relates to the blood vessels around it, where it gets its blood from, where it's sending blood to, and so on and so forth. Right? Also, remember, left and right matters. Right? If you're being wheeled into surgery at the emergency room because you have a cut in your femoral artery, do you want your doctor to just flip a coin to see which one he is going to fix? No, you definitely want to know left and right. In your textbook, Christopher, I don't have any of the illustrations, I don't think readily available myself, but here we go. If I can find one real quickly.
738, I think.
Yeah, perfect. Yep, that's a good one. Here's another good one. I'll just show this one. Stop sharing for a minute. So notice associated with the arm you can see that on one side of the book, they have the actual arm and the chest and things like that. But notice on the other side, they just have this simple schematic showing how they relate to each other. So these are things that could definitely be useful as you are learning these. I obviously won't show you one of those schematics to hold you responsible for what is this blood vessel in the schematic, but could I show you the first image of the chest and ask you, or the arm and ask you? Absolutely. So, and remember, your textbook, your atlas, your lab manual, all of those things show far more blood vessels than you're responsible for. Remember, you're only responsible for the blood vessels on your list. So, as we talked about when we're talking about these systemic blood vessels, we have that oxygen-rich blood that leaves the left ventricle and passes through what structure?
The aortic valve.
Yeah, the aortic semilunar valve. Excellent. And it enters into what blood vessel?
The aorta.
The aorta. Excellent. What image, just out of curiosity, did they have that flipped? Ash? The neck. Oh, interesting. Cool. All right. Aortic semilunar valve, and it goes into the ascending aorta. Remember, we are meaningfully dividing the aorta into 4 different divisions. So when you're giving your driving directions, don't just say aorta. If you're going to the leg, for instance, you've got to go through the ascending aorta, you've got to go through the aortic arch, you've got to go through the thoracic aorta and through the abdominal aorta. And the reason we divide it into those 4 meaningful pieces is because of where they distribute blood. The ascending aorta distributes blood to one and one structure only. What is the one and one structure only that receives blood from the ascending aorta?
Aortic arch.
Well, okay,
Coronary
so
arteries.
goes to the aortic arch, but what part of your body?
The heart.
Heart. How many blood vessels come off of the ascending aorta?
2.
2. What are they?
The right and left coronary artery.
There you go, exactly. The only thing that gets blood from the ascending aorta, or if you want to think about it, the very first thing that receives oxygen-rich blood as it leaves the heart is the heart itself in those coronary arteries. And here in these coronary arteries are where we see our first major asymmetry. Notice, as we see here, and look, thankfully they've removed that pesky pulmonary trunk for us so we can truly see the right and left coronary arteries. We can see how the right coronary artery travels in this indentation between the atria and the ventricle. What might that indentation be known as?
Coronary sulcus.
Nope, although that's a good guess, but let's be more specific. It is a sulcus, but what sulcus would it be between the atria and the ventricle?
Atrioventricular sulcus.
There you go, just that simple. The atrioventricular sulcus is that indentation between the atria and the ventricle. Notice as it travels around in that atrioventricular sulcus, there is a major branch that comes off that goes along the lateral aspect of the heart, or the right lateral aspect of the heart, known as the marginal artery. So the marginal artery goes along the margin of the heart on the right side. And then it continues around to the back. Notice as it continues around onto the back, here we see it again, coming around all the way to the posterior side of the heart. It then goes down this indentation located between the ventricles. What would this indentation between the ventricles be known as?
The intraventricular sulcus.
There you go, the interventricular sulcus. Absolutely, excellent. And what is the artery that sits in the interventricular sulcus?
Posterior interventricular artery.
There you go. Isn't that convenient? The posterior interventricular artery is in the interventricular sulcus on the posterior side of the heart. So notice our right coronary artery is very long and travels all the way around to the posterior side of the heart. So does that mean the left one does the same thing on the other side?
No.
No. If I'm making a point of emphasizing it, then absolutely the answer is that it doesn't. And so here we see that the left coronary artery is actually very, very shy. It is small and basically is almost completely behind the pulmonary trunk. Notice if the pulmonary trunk was in place, it would basically be hidden almost entirely by that pulmonary trunk. Instead, it pokes its head out and branches almost immediately. When it branches, one of those branches travels down the indentation between the ventricles on the front of the heart. What would that indentation between the ventricles on the front of the heart be?
The anterior interventricular sulcus.
Yeah, and what is the artery that sits in the anterior interventricular sulcus?
Anterior interventricular artery.
Excellent. And notice in the anterior interventricular sulcus, there is also a vein. So if it's the anterior interventricular artery in the anterior interventricular sulcus, then what, of course, would the blue blood vessel, the vein, be in that anterior interventricular sulcus?
Anterior interventricular vein.
That would make a lot of sense, but what do we know about anatomists?
They hate us.
ATAS.
So what did they call it?
Great cardiac vein.
Yep. Right? Why? Because they hate you. I can't think of another reason why they would do this. The only reason they would possibly do this is because they truly hate you. Now, this brings us back to another point I have said many times, and will continue to say many times, is that people often lose points not because they don't know the information, but because they don't read the question carefully. Notice on the exam, I could have a picture just like this, or a model just like this, with an arrow pointing just like that one. And notice there are 3 questions I could ask. Identify the indentation indicated by the arrow. And of course, what would your answer to that be?
Anterior interventricular sulcus.
I could ask you to identify the red blood vessel indicated by that arrow, and that would be the
anterior interventricular artery.
Or I could ask you to identify the blue blood vessel indicated by this arrow, and your answer would be
Great cardiac vein.
Great cardiac vein.
exactly. People often get really excited. Again, these lab exams can be exhausting. So you look at a slide, you see the arrow, you get all excited. I know that that's the anterior interventricular artery. But make sure you read the questions carefully. Again, my goal is not to be tricky. So I'm telling you all of this right now, but we do have to be specific. I will be very precise with my wording of the question. I expect you to be equally precise with your answer to it. Because remember, there's all those other questions too. I could ask you the condition of the blood in the red vessel. What would your answer to that be?
Oxygen rich.
Yeah, I could say identify the blood vessel that feeds blood directly into this blood vessel. And what would your answer to that be?
Left coronary artery.
Right. So again, there's easily a half dozen questions or more that I could ask with this image and that arrow right there. Read the questions carefully so you can answer them correctly. I've not heard the term left anterior descending. That's not a term I'm familiar with. As I've said before, there are often many multiple appropriate terms for blood vessels. If you have a picture in your textbook that refers to that as the left anterior descending, and you showed me that because I marked it wrong on the exam, then I would give you credit for it. But that is not a term that I'm familiar with, left anterior descending. But again, cardiovascular is not my area of expertise, but that is not a term I'm familiar with. All right. Notice that's just one of the paths, that one of the branches that comes off the left coronary artery. The other one is the circumflex, right? Like it goes around the circumference of the heart on the left side between the atria and the ventricle. So again, in that atrioventricular sulcus. And here we see that circumflex artery coming all the way around to the posterior side. All right. Questions on that? Yes, both lab and lecture exams will, all the questions will be available all at once. And again, that's why I said many people who have had problems, not a lot of people have had problems. Proctorio and Canvas has gotten better over the past year. But the people who do have problems, the rare few people who do have problems, tend to have problems more on the lab exams where you're loading 80 images as opposed to just the lab and lecture exam where you've got, 30 multiple choice questions and, 12 essay questions or something like that. All right, we've gotten blood to all the parts of the heart. Let's get the blood back from all parts of the heart. And not surprisingly, where you find the arteries is where you find the veins. We already mentioned the great cardiac vein, which is great because it's on the front, but it's also the longest, and that's why it gets that name great. Notice it travels up the anterior interventricular sulcus and then around the atrioventricular sulcus into the back of the heart. Notice the same place where the marginal artery is where the small cardiac vein is. And the way I always remember it is if you do something to try to marginalize someone, you make them feel very small. Of course, if we have a small, we need something in between. What's that something in between going to be? Well, what's between small and great?
Probably medium.
Yeah, right. Middle cardiac vein, absolutely. And notice it is the middle cardiac vein that travels up the interventricular sulcus on the posterior side. However, remember, one of the things we know about the heart is that it has a thicker left muscular wall. So on the left side, we need larger muscle, we need more blood. And so notice there is a larger vein that drains blood from the left ventricle that is known as the posterior cardiac vein and is actually sometimes literally referred to as the posterior cardiac vein of the left ventricle. You don't have to use all of that in its title. You can just refer to it as the posterior cardiac vein. But you, in the literature, may also see it as the posterior cardiac vein of the left ventricle. Now notice all of these blood vessels, the small cardiac, the great cardiac, the middle cardiac, the posterior cardiac, all of these veins feed into one big, huge, loose blood vessel. One big, huge, loose vein. And we have a special name for a big, loose vein that is able to collect a lot of blood. What do we call a big, loose vein?
Sinus.
Sinus, exactly. And so it is this coronary sinus that collects all the blood from the heart and, as we know, is one of the 3 blood vessels that feeds blood into the right atrium. And of course, what are the other 2 blood vessels that feed blood into the right atrium?
Superior and inferior vena cava.
There you go. Superior vena cava, inferior vena cava. Perfect. So there is our heart. The vascularization of the heart, somewhat unique, not surprisingly. And again, our illustration does a decent job of showing it. It's even more so. Massive numbers of anastomoses. We want all these interconnected blood vessels because we want to make sure there's lots of different routes to make sure that blood gets where it needs to go. Because if blood got blocked in one of these areas, right, that could cause problems. Now the blood's not getting to a location. Luckily, we can do a surgery where we would actually go in and take a blood vessel off of a cadaver or maybe a blood vessel out of their leg or something like that, where we could reconnect the blood vessels across that blockage. And what would we call that type of surgery?
Bypass graft.
Yeah, bypass surgery, exactly. Again, one of the things we're missing out by not being in the classroom, one of the hearts we have to study from our cadavers actually has 4 bypasses on it. So again, we don't have all the medical information. We do not know if they got all 4 bypasses at one time or if it was over multiple surgeries or whatever, but we actually have a heart that actually has not 1, not 2, not 3, but 4 bypasses on it. It's pretty cool looking. Excellent. Yeah. Again, the practice anatomy lab is a great way to study the material. And remember, it's got the quiz and the exam where you can actually test yourself on that information as well. Now, remember, and if it's pointing at a blood vessel that's not on your list, ignore it and move on to the next one. I don't see the scores or anything from that. So again, no pressure on that. Use it as a great tool. All right. We did all that. We did all that. We did all of that. Excellent. So that is the ascending aorta. So if we don't leave the coronary arteries, we go from the ascending aorta where?
The aortic arch.
Aortic arch. How many different regions of the body does the aortic arch feed?
3?
3? Well, what are they? Name them.
I believe it's the right and left clavicular and the common carotid.
Regions of the body.
Never mind, count me out.
Well, you have the right information. And again, this is one of the, your information is correct, but again, you didn't answer the question. You got to be careful of that. What are the, give me one region of the body that is fed blood by the aortic arch.
Isn't it the lower body and the left side of the arm and shoulder?
One specific region of the body. If you put upper part of the body, is that a region? Lower part's not a region of the body. Think back to your regional terms. What is one region of the body that I'm waving through space that receives blood from your aortic arch?
Extremity.
How about be specific? How about the right arm? Give me another one.
The left arm.
There you go. Give me another one.
The head.
Okay, but does the head just get one main blood flow? Or is it the right side of the head and the left side of the head like we saw? So right arm, right side of the head, left side of the head. 4 different regions of the body that get blood from the aortic arch. So of course, how many branches does the aortic arch have coming off of it?
4.
Exactly. Right. So here we look at the aortic arch and see 1, 2, 3, crap.
I knew it.
4 regions of the body that get blood from the aortic arch, but there's only 3 branches that come off of it. What are those 3 branches? What's this one here called?
Left subclavian.
Guess where it goes?
Left side.
Underneath the left clavicle.
Okay, but ultimately goes to the left arm, right? We were just talking about that left arm. Left common carotid artery. Guess where it goes?
To the head.
Left side of the head. What's the 3rd blood vessel called?
Brachiocephalic trunk.
Brachiocephalic trunk. 2 important things here. First, trunk. And we know a sinus is a large loose vein for collecting a lot of blood. Anybody know what a trunk is?
A segment that branches immediately after.
Excellent. It is typically a short artery that branches almost immediately. Excellent, exactly. And this one happens to be the brachiocephalic trunk. Brachio refers to what?
Arm.
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 Cephalic refers to what?
Head.
Head. So notice this particular trunk is going to branch quickly, and it is going to feed both the right arm and right side of the head. Right? We don't have a left head and a right head. We have a right side of the head and a left side of the head. So stick that in there. So there you go. That's how we get away with that. Now, again, this is a weird asymmetry. Why do we need it? Why not just have 4 blood vessels coming off the aortic arch? Well, what do we know about the heart? Is it located smack dab in the middle of my chest? No. Remember, why is one of those questions that's hard to answer in anatomy and physiology. Again, the engineer who designed this didn't leave us her footnotes. But if I was a plumber and I had to lay pipe from one location to another, and I had my pump that was slightly to one side of the house versus the other, wouldn't I need a little extra tubing to get to the right? Yeah, and that's exactly what happens here. Right here to get to the right, we have this brachiocephalic trunk, which, as we know, is going to branch almost immediately. And when it branches, guess what 2 branches it forms?
The right common carotid artery and the right brachial artery.
Not
right subclavian.
Good, excellent. And the right subclavian. After that one brief asymmetry, basically everything is the same left and right after that. So the one asymmetry we have is this brachiocephalic trunk. The right common carotid, the right subclavian come off of the brachiocephalic trunk, whereas the left common carotid and the left subclavian come off of the aortic arch. But after that, they all lead the same places. So let's take a look at that. If we are going to notice this route is going to the right, but it's exactly the same to the left. If we come off of the brachiocephalic, we go into the right subclavian, which, not surprisingly, as the image shows, goes under the clavicle. To the blood vessel through the armpit, which is, of course, the
axillary.
All right, which goes to the one of the upper arm.
Brachial.
Which goes to the 2 primary ones of the lower arm.
Radial and ulnar.
Right, radial and ulnar, and eventually to the deep and the superficial palmar arches. Right? This path is exactly the same to the left. The only difference would be on the right, we have to go through this first, whereas to the left, we do not. We go straight from the aortic arch to that. So when you're giving drive-in directions to your right thumb versus your left thumb, right, there's only one difference between those 2 pathways. Same thing to the head. Again, to the right, we have to go through the brachiocephalic first, but then we go into the common carotid. That common carotid then branches into both the internal and external carotid. The external carotid basically comes superficially to the skull, to the muscles, to the skin, whereas, as we talked about, we know that internal carotid goes up to the brain. Notice, remember, we mentioned those 4 blood vessels that feed the brain, the 2 internal carotids and the 2 vertebral. Notice here we see that vertebral artery going through those transverse foramens up into the back of the brain. And notice it actually comes off the subclavian. So off of the subclavian, we have our vertebral that again feeds into that circle of Willis we talked about. Notice there's one more blood vessel that comes off here we'll talk about as well, but we'll talk about that one later in just a minute. We already did the circle of Willis. So again, I'll just emphasize again, Here are where the two internals are coming up. Here are where the two vertebral are coming up. And we already did the rest of these, so I'm not going to waste time going through them again. But we did all of that. All right, excellent. And then again, left common carotid, left subclavian, do the exact same things. So that brachiocephalic is the only asymmetry from the left to the right. All right, I think that's pretty straightforward. Like I said, I don't have time to go through all of these. I want to deal more with the distribution of the blood and also these. I don't know if these slides are on Canvas, but almost all of these pictures are straight out of your textbook. I know I have unlabeled pictures on Canvas that you could write these labels yourself. So I do have that available for you. But most of these pictures I pulled straight out of your textbook. All right. All right, excellent. So again, from the aortic arch, we go to that descending aorta, which is both posterior and inferior of the heart. But remember, the key to that descending aorta is that it passes through the diaphragm. So it's much more meaningful to talk about what is above the diaphragm and what is below the diaphragm. Because again, we want to worry about the distribution of the blood. And so we distinguish the thoracic aorta versus the abdominal aorta. So those are the four, and again, I hate to use the word branches, so 4 segments, let's say it that way, 4 segments of the aorta that we want to distinguish. Ultimately, when the aorta finally terminates, it branches into 1, 2, 3 blood vessels. And notice we look closely, this particular illustration doesn't show the third, but I'll ask it anyway. Anybody know the 3 blood vessels that the aorta, when it terminates, finally branches into? You can start with the 2 easy ones if you want to start with those.
Common iliac artery.
Excellent. And right and left common iliac. So those are 2 of them. Anybody know the third? If only I had a list of all the blood vessels I was responsible for in this class, I may be able to infer what the correct answer would be to this and then make myself look very impressive in front of all the other students. Still waiting.
Mesenteric artery.
Not a bad guess, but nope. If only I could visualize what this triangular region down here in the inferior part of my pelvis was known as.
Medial sacral artery.
There we go. Excellent. I appreciate the dramatic pause to make everybody stress about what that was. Excellent. Median or medial sacral artery, which basically feeds the sacrum and coccyx. Excellent. We'll take a quick look at that in a minute. But before we get there, we need to sneak past the thoracic cavity. Remind me again what the thoracic cavity contains.
Lungs, heart.
Lungs and heart. Do the lungs have their own blood supply? Maybe one might even call it the pulmonary circulation because it goes to the lungs. Does something like that exist? Yeah, there you go. Does the heart have its own blood supply?
Yes.
Excellent. Right? So notice in that case, we don't have any really major organs that require blood supply from the thoracic aorta. So there are no major branches coming off. Notice the primary branches come off are going to the bones and the muscles of the thoracic cavity, the bony thorax. So notice we have our posterior intercostal arteries coming off of the thoracic aorta. passing in that indentation in the ribs, which I'm sure you remember is that costal groove where we said the blood vessels and the nerves would sit. However, remember up here off of our subclavian, we talked about how we had that vertebral artery that goes up. Well, notice there's also a blood vessel that comes down the front of the thoracic cavity that also comes off that subclavian. Any idea what we might call such a blood vessel that comes off of the subclavian down the front of the thorax?
Internal thoracic artery?
Yeah, it's the internal thoracic artery. And notice it feeds into the blood vessels that go onto the anterior intercostal arteries. So we have blood going to the front and blood going to the back of the ribs from these two main sources. But notice there's not a whole heck of a lot going on in the thoracic cavity because the heart and the lungs have their own major circulations already associated with them. Where things get fun is in the abdominal cavity. Now notice there are a lot of blood vessels, both paired and unpaired, that come off of the abdominal aorta. Notice also, as we just finished talking about, when the abdominal aorta terminates, it terminates into the two common iliacs. And here we see that median sacral artery feeding the sacrum and coccyx. But notice there are a large number of blood vessels that come off of the abdominal aorta, both paired and unpaired. So this is one of those cases where I think it might be useful to actually draw this. There is actually an illustration I think I found, or a schematic that I found that I posted for you on Canvas. Here is our abdominal aorta. And of course, we have our diaphragm up here at the top. And then as we know, this is going to branch into those three blood vessels, the right and left iliac, common iliac, and the median sacral. All right, so from our diaphragm down, the first blood vessel that comes off, is it a paired or an unpaired blood vessel?
I'm sorry, can you say that again?
The very first blood vessel that comes off of the abdominal aorta inferior to the diaphragm, is it a paired or an unpaired blood vessel?
Unpaired.
Blood vessel, or is it a paired blood vessel?
A trunk.
Unpaired.
There you go. In fact, it is indeed a trunk. It is a single unpaired blood vessel. And what trunk is it?
Celiac trunk.
We know trunks are small, short blood vessels, well, large short blood vessels that branch almost immediately. And how many branches does our celiac trunk have?
2.
Maybe. 3. There you go. 3 branches that come off of them. What are they?
Common hepatic artery, splenic artery, and left gastric.
Common hepatic. Where does that go?
Liver.
Liver. What was the next one?
Splenic.
Splenic. Where does it go?
Spleen.
Spleen. And what's the third?
Left gastric.
Where does that go?
Stomach.
Yep, and more specifically, the left side of the stomach. Does that mean that the right side of the stomach doesn't need blood?
No.
Nope, it needs blood. Anyone know where the right gastric comes off of? Comes off the common hepatic. Right gastric going to the right side of the stomach comes off the common hepatic. So there are 3 major branches that come off of that celiac trunk, going to 3 different destinations. Excellent. What's the next blood vessel on our list that we're responsible for on our way down?
Superior mesenteric.
Single or paired or unpaired?
Unpaired.
Unpaired. Excellent. So I have a single blood vessel. And what did you say it was called?
Superior mesenteric artery.
Any idea where that goes?
Goes to the
small
mesenteric.
intestine.
Excellent. It goes to the mesenteries, and the mesenteries go to all of the small intestine and the proximal large intestine. Excellent. What's next? As we work our way down.
Renal artery.
Paired or unpaired?
Paired.
Paired. So we have the paired renal arteries. And where do they go?
Kidneys.
There you go. All right, we have the paired renal arteries going to the kidneys. As we talked about while you're sitting here at rest, about 25% of your blood is being directed to your kidneys from that. What's next?
Gonadal artery.
Paired or unpaired?
Paired.
Excellent. Grant, where do they go?
Testicles and ovaries.
There you go. Testes and ovaries. That means there's going to be some differences in these because the testes and ovaries are not the same. Which one is longer?
Testes.
Testes.
Yeah, testicular. Arteries are longer. Why?
Is there lower?
Farther to go.
Yeah, got further to go, right? The ovaries are located in the abdominal pelvic cavity, right? The testes are located outside the abdominal pelvic cavity, inferior to the abdominal pelvic cavity. Which ones are larger?
Ovaries.
The ovarian?
Ovarian arteries, yes.
Well, why would you need it to be larger?
Because the organs are larger.
Okay, actually the gonads and the testes and the ovaries are actually very similar in size. Why might you need a larger blood vessel just going to an organ in general?
More blood supply.
More blood
more
supply,
nutrients?
more oxygen, more nutrients, more metabolism. Whose gonads are more active, males or females after puberty?
Females.
Females? How many gametes does a female make during the course of a month?
Not millions.
0.
Females already have their eggs, so they don't have to keep making new ones.
Exactly. Females, being the overachievers that they are actually born with all the gametes they will ever need. Now, during the course of a month, they need to mature gametes, but during the course of a month, how many gametes actually reach maturity for a female?
1.
1. How many sperm have I made while I've been sitting here giving this lecture?
Jillion.
Exactly. Jillions of them. Absolutely. Right? Males are so busy making gametes. Quite frankly, I don't know why we're required to do anything else. We should be able to just sit at home and make gametes, and that should be our job. Right? So not surprisingly, from a metabolic standpoint, there is a much more need for energy, for oxygen, for resources. So it is the testicular that are both longer and larger. All right. Excellent. What's next?
Inferior mesenteric artery.
Inferior mesenteric. Single Paired? Which one?
Single?
Single.
It's unpaired, absolutely. I guess you could argue it's paired because we have a superior mesenteric and an inferior mesenteric, but they're each one is unpaired. This inferior mesenteric artery basically goes to the distal portion of the large intestine. Right? The sigmoid colon, the rectum, the anus. And so these are the blood vessels that come off of the abdominal aorta. Notice if we go back to the pretty picture from your textbook, we can see this very clearly here. We have our celiac trunk, superior mesenteric. Purple doesn't show up very well. Celiac trunk with its 3 branches, superior mesenteric, paired renal, paired gonadal, unpaired inferior mesenteric, and then it finally terminates at the end. Now notice this just shows the branches coming off of them, but notice if we put in all the abdominal hunk and junk, here we can see not just the 3 branches that come off the celiac, right? The left gastric, the splenic, which is coming over this way, the common hepatic, but notice also off the common hepatic, we can see that right gastric that comes off of it. We see the superior mesenteric going to the small intestine and the proximal large intestine. And we see the inferior mesenteric going to the distal part of the large intestine. So here we can see kind of those different branches and where their actual destinations are as they're going through this. Allison, I would post my drawing, but I believe.
Or maybe if you can go back and I can snap a shot of it.
Well, here, hold on. Let me wait, because I thought I had kind of already done this. Let me get to the right place. Are we on time? We're still okay. All right, so when you go to the modules, under lab handouts, notice we have some unlabeled blood vessel pictures. So for those of you who are looking at these or furiously taking your snapshots or whatever, again, I have all these pictures of some of the different images that we've looked at where you can actually do your own labeling of some of these things. But the other thing that I put in here, and I'm happy to post my picture, but I thought, oops. Notice this is a little nicer than mine, and it also shows the veins at the same time that it shows the arteries. So we have this image here as well as the images in your textbook, and this one also shows the veins that we're going to be talking about as well. So I'm happy to post my picture, but there are these other images there as well. So actually, while we're thinking about it, let's go back to my whiteboard, and I'll go ahead and save this image so that we have it. I can post that. All right. Questions on that? All right, excellent. From here, we did all of that. From here, we get to the legs. Again, no difference in left and right other than identifying left and right. Much like we have the common carotid that branches into the internal and the external carotids, we have our common iliac, which branches into the internal and the external iliac. Internal iliac feeds many of the organs in the pelvis, a bladder, uterus, things along those lines. The external goes to the leg, feeds into the femoral, feeds into the popliteal. And notice, remember, as we talked about, we call this the popliteal artery, and there is indeed a thoroughfare that passes through here. But really, all of this mesh, this huge anastomosis, is kind of all considered to be part of that right popliteal artery, because again, we want that anastomosis, so no matter how you bend your knee, blood can get through. All right, then anterior, posterior, lateral is the fibular or perineal. Remember, fibular and perineal are interchangeable terms. So again, I think, like I said, there's no real tricks to the lower appendages on the way there. All right, and complete symmetry from the two sides. Now that we've gotten blood to all parts of our body, we need to get them back from all parts of the body. And remember, as we talked about, there are more veins, more passageways. The veins are a little bit trickier. One of the places where the veins are the trickiest is in the skull, because we want to make really sure that we're draining all the blood and all the cerebral spinal fluid out of our cranial cavity, right? Yeah, I want to drain all the blood from my hand as well. But if I get a little bit of edema in my hand, right, the worst thing that happens is it makes it a little harder for me to get my wedding ring off when I get to the bar on Friday night. All right. However, do we mind if there's a 20–25% swelling inside of our cranial cavity?
Yes.
Yeah, because unlike this part of my body that can swell to accommodate that, we're locked inside of a big locked cranium. And that swelling around the brain can be incredibly dangerous. Now, vodka is strictly a morning drink. At the bars, I always order a Long Island iced tea because the ladies love that. All right. All righty, excellent. Exactly. Or if I'm feeling really fancy, a Cosmo, maybe a dirty martini. All right. Go with my necklace and heels. All right. Exactly. So we want to make sure we are doing as much as we can to drain the blood out of the cranial cavity. And so we have a large number of sinuses. Remember, those sinuses are big, loose blood vessels for draining space. The first one you should be familiar with because you learned about the falx cerebri in the 430 class. Anybody remember what the falx cerebri was again? If only I had paid attention in 430 and my instructor was talking about the nervous system, especially if that instructor was Daniel Slutsky, who loves the nervous system so much. Anyone remember what the falx cerebri was? It's so weird the way my microphone seems to cut out occasionally when I ask questions in this class. If you remember correctly, they're extensions of the dura mater that help to support and stabilize your brain and hold it in space. One of those was that falx cerebri, where the dura mater extends down into the longitudinal sulcus, helping to stabilize the two hemispheres of the brain. So right on the midline where that dura mater, the first layer of the dura mater comes off and goes down, is this big, large, loose sinus right at the top of the cranial cavity, known as the superior sagittal sinus. And anytime they call something a superior something, you know that they are also going to have an inferior something. And that inferior sagittal sinus is a slightly smaller one, deeper in the falx cerebri. Notice the inferior one is small enough where it doesn't fully go all the way to the back. Now we'll talk about that in a second. So it feeds into the straight sinus, which is also on the midline. And then when those two fuse together, they form this S-shaped sinus known as the sigmoid sinus. Now, the other thing you may remember from 430 is, remember, in our superior sagittal sinuses, that is where we drain our cerebral spinal fluid. Remember, our cerebral spinal fluid circulates through the ventricles, circulates around the spinal cord, circulates in the subarachnoid space. And then those are those finger-like extensions of the arachnoid matter called those arachnoid granulations that feed the cerebral spinal fluid back into the superior sagittal sinus. So again, not only are we draining the blood from the brain, cranial cavity, but we're also draining the cerebral spinal fluid from the cranial cavity. Now notice all of these structures are on the midline. And even though this picture doesn't really show it, there are two sinuses that go along the lateral aspect of the cranial cavity. What do you think those might be referred to as?
Lateral cranial sinuses.
Lateral would be good, but like I said, if I would cheat and have my study guide in front of me so I could be reading off the list, lateral is a good term, but also from the side would be, or because
Transverse?
Transverse sinuses, exactly. Those transverse sinuses come together as well. So I don't know if I have a picture of that. I don't, but they are lateral and they also feed into the sigmoid sinus. This sigmoid sinus is one of the primary, largest of the sinuses, and its job is to collect most of the blood from the brain and the cranial cavity. Notice we have this deep cavernous sinus, which helps to drain, like for instance, the eyes and the anterior part of the brain. And notice these all feed primarily into the internal jugular. Huge asymmetry between the internal and external jugular. The internal jugular is way, way larger because its job is to drain all of that stuff out of the cranial cavity. Right. Again, we have vertebral veins that help with that as well. And notice the external jugular primarily comes from superficial blood vessels. Now, again, normally we have symmetry: brachial artery, brachial vein, carotid artery, carotid vein. No, the head is one of those areas where we have a lot of asymmetries, right? And one of them is carotids, arteries, jugular veins.
Question, do all the veins still have those valves, or because gravity's helping here, it doesn't bother with the valves?
Sinuses typically don't have valves, but the blood vessels, I would say, do, because while you're correct, you do have the advantage of gravity helping in this case. Again, it is so important to get blood out of the cranial cavity. I would say that the veins, smaller veins, do indeed still have valves in them. Yep. Now, notice both the internal jugular, and in this case on the right side, the subclavian and the external jugular, all kind of come together to form a brachiocephalic vein. How many brachiocephalic arteries did we have?
One.
One, so we don't have to worry about right or left. How many brachiocephalic veins do we have?
Two.
Two.
So here we do have to worry about saying right or left. Now, why do we have two brachiocephalic veins and one brachiocephalic artery? Again, I don't know, but that is the way it is. So the right side and the left side for the venous structures are completely symmetrical in how they drain. There's no asymmetries at all. Both have brachiocephalic veins that receive blood from the internal jugular and the subclavian to form those brachiocephalics. And notice the external jugular feeds into the subclavian before it feeds into the brachiocephalic vein. Of course, the two brachiocephalic veins come together to form what blood vessel?
Superior vena cava.
Superior vena cava. So notice the superior vena cava drains the right side of the head, the left side of the head, the right arm, the left arm, And as we'll see, it also drains the thoracic cavity, but not from anything we see right here, but we'll get there eventually. All right.
So all of the veins on our list from the brachiocephalic down to the transverse sinus, they're left and right?
The inferior, superior, and sagittal, so most of them are paired. Inferior sagittal, superior sagittal, the straight sinus, the sigmoid sinus, and the cavernous sinus. All of those are unpaired. The only ones that are paired are the transverse sinuses. So you have a left and right transverse sinus, but all the rest of these sinuses are on the midline. So there's only one.
Straight is not paired, right?
Correct. Yeah, the sigmoid sinus feeds both internal jugulars. And so that is where it becomes paired coming out. All right. Again, I think with the veins, the head is one of the trickier areas. Arms, much easier because there is tremendous symmetry. We have brachial arteries, brachial veins, axillary arteries, axillary veins, so on and so forth. However, there are a couple blood vessels that are different in the arm, vein-wise. We have, for instance, this one here that goes across the arm pit, the elbow joint. Anyone know what that one's called?
Antecubital vein.
Well, actually, medial cubital vein. And why is that one significant? Why may I make a point of pointing that out?
It's where we get blood draws and IVs.
Yeah. Typically, when you draw blood, that is where you draw it from. Interestingly, there are two other blood vessels. There is the brachial, notice, is deep, but there are these two superficial blood vessels. One is on the lateral side of the arm, and one is on the medial side of the arm. So of course they have names based on those locations. And what do we call this lateral superficial blood vessel of the arm?
The cephalic vein?
Right, because of course cephalic means lateral, right?
No, it doesn't.
What does it mean?
It means head.
wait. Why do we have a blood vessel called head vein in our arm?
That's a good question that I cannot answer.
Well, luckily I can.
Anatomists hate us.
That is part of it. It's because anatomists hate you, exactly. But their rationale from it is from a developmental standpoint. When we are developing as little embryos and fetuses inside of our body, as the arm bud forms, the arm bud is actually up above the head. And so this blood vessel on the lateral aspect of the arm is right next to the head. So that is called the cephalic vein, whereas the one that is away from it, kind of the base of the arm bud on the other side, is then referred to as the
Basalic.
Basilic, exactly. Notice not basilar. Basilar was the one in the skull. This is basilic, not basilar, basilic. Yes, anatomists are horrible, horrible people. That was the one thing I was excited about when Trump was president. He was so anti-science. I was really hoping he was going to take all the anatomists out and shoot them. But couldn't even do that right. All right. So again, weirdness. So again, remember first of all, be careful. They don't come off the subclavian because remember, veins are feeding blood back towards the heart. So really it's the other way around. These blood vessels, the basilic, the cephalic, and the brachial merge together to form the axillary. So they're merging together to form into the axillary, and then the axillary feeds into the subclavian. But notice, even our artist illustration, there's all these anastomoses. So it's a very intertangled web of blood vessels that we have here. But they feed into the axillary, which ultimately leads into the subclavian. And again, perfect symmetry in the arms. One place we don't have symmetry is in the thoracic cavity. Notice in the thoracic cavity, again, the heart and the lungs have their own circulations we don't have to worry about. But we had all that blood that was feeding into the muscles and the ribs. And so notice all of those intercostal veins feed into this single blood vessel on the right-hand side that feeds into the superior vena cava. And that right side blood vessel is the azygous vein. Now, shouldn't it be the right azygous since it's on the right side?
There's only one.
Exactly, there's only one. Because what happens is the left side tried to work the same way. The left side was on board for a while. All right, I'll drain all those blood vessels from the ribs as well. But then the left side got tired, got lazy. I don't want to have to carry the blood. It's kind of like my teenagers. I don't want to have to carry the blood all the way back to the superior vena cava. So I'm just going to feed it to the azygous and let it do its work. I'm only going to take it halfway. And since I'm only going to take it halfway, that inferior blood vessel on the on the left side is the hemiazygous, because of course hemi means half. Exactly. Notice that doesn't help us for the superior part of the left part of the thoracic cavity. So notice the hemiazygous needs help. So the hemiazygous needs help from the superior part. So there is a superior blood vessel that helps the hemiazygous, known as the accessory hemiazygous, right? Because it takes two teenagers to do the job of one mom, right? And that's what we have going over here. Mom's on the right, kids are on the wrong, on the left. And so we have the hemizygous, the accessory hemizygous, and we have the azygous. 3 blood vessels draining blood from the thoracic cavity. But notice only one of them feeds them into the heart, and it does it by feeding it into the superior vena cava. In fact, if you look at your models and stuff, which you should be looking at, and this is actually a good reminder of that, let's quickly see. I haven't actually looked at this myself, but let's see what's there. Go back here. Remember, if we go to our modules and we go to that Cosumnes River College Virtual Anatomy Lab, and we scroll down to hearts, see if we find one that has a good view that I want. No, 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 no. Where's the back of these? There we go, that's not bad. Okay, this is not a good view. This would not be the view that I would use on the exam, because obviously they're trying to show us the coronary sinus and the middle cardiac vein and the posterior vein of the left ventricle and all that. But if you look closely, again, I would not use this illustration for this view on the exam, for this question on the exam. But if we look closely, notice this is the right brachiocephalic vein feeding into the superior vena cava. And notice this little blood vessel feeding into the superior vena cava from the back. That's the azygous vein. So notice we can actually see this azygous vein as this little blood vessel that feeds into the back of the superior vena cava. You got to be looking at the back of the superior vena cava to see that. Let's see if they've got a better view. No. Slackers. Okay, they don't have a good view of that, but I'm sure if you use your personal anatomy lab, you can see some posterior views and see a nice view of that. So again, I would not use this on the exam, but this is the one example from Cosumnes River. And notice they haven't even bothered labeling all of the stuff up here. But oh, let's see, look at some other cool things we can see here. Notice this is our descending aorta, our thoracic aorta. Notice we can actually see those posterior intercostal arteries coming off of the thoracic aorta. And as we mentioned, we have that azygos feeding into the superior vena cava from back here. And then, of course, this is the esophagus and so on and so forth. So, yep, the zygos feeds into the superior vena cava. That is correct. All righty. Here we see it as well. Notice here it kind of shows it from the side. It's really from the back. All righty. We did that, we did that, we did that. Perfect. Notice when we look at the abdominal cavity veins, our inferior vena cava is not nearly as busy as our abdominal aorta is. Notice we have some we recognize: renal arteries, renal veins. At the end, it terminates, or I said it should form, from the common iliac veins and the median sacral vein coming together to form the inferior vena cava. But no celiac. Nothing from the stomach. No mesenterics. Nothing from the superior and inferior mesenteric. Notice what we do have: a bunch of blood vessels that are hepatic veins. Where are they coming from?
Liver.
From the liver. What about gonadal veins? How many of those do we have?
2.
2. And do they both feed into the inferior vena cava?
No.
No. Notice the right gonadal vein feeds directly into the inferior vena cava, where the left gonadal vein actually feeds into the renal vein first, and then obviously from there into the inferior vena cava. Now, again, some of these asymmetries from a plumbing standpoint, we can kind of understand or interpret and make sense of it. I will tell you with 100% certainty, I have no idea why this asymmetry is here. It makes no sense whatsoever, but it is a consistent characteristic, but we have no idea why. All right. But no mesenteric, no splenic, no stomach, right? Nothing from the large intestine, from the small intestine. Why? Notice there are no veins from the digestive system directly to the inferior vena cava. So the question becomes why?
They feed into the liver first.
Exactly, absolutely. All digestive veins feed into the liver first. Why?
Because
Filter.
they're drawing nutrients and such from the intestines, and the liver needs to metabolize certain toxins. And
yeah, part of it is toxins, but part of it is also there is a difference between being nutrient rich and being nutrient appropriate. While I know every single person in this class does a very good job with their breakfast, making sure they have the direct amount of magnesium and calcium and carbohydrates and amino acids and proteins and all of those types of things in their breakfasts in the morning, there are some people that will have a donut and a Diet Coke for breakfast. And if you're having a donut and a Diet Coke for breakfast, are you necessarily getting all the nutrients that you need? No, you are not. You may be getting some, but you're not getting, so it may be nutrient rich. It's going to have a lot of glucose in it, for instance, but it's not going to be nutrient appropriate. So what has to happen is all of this blood has to go first to the liver. And because we're not talking about the digestive system yet, I can tell you with a completely straight face that in the liver, magic happens. And that magic that happens makes the blood nutrient appropriate. And once it is nutrient appropriate, then it can go back to the heart, and it goes back to the heart via the hepatic veins. Now, to get to the liver, for that magic to happen, all of the digestive veins have to feed into a blood vessel that goes to the liver. And anyone know what the name of that blood vessel that goes into the liver is?
Portal vein?
Exactly, the hepatic portal vein. The hepatic portal vein receives the blood from the digestive system and feeds it into the liver. The hepatic veins take it from the liver into the inferior vena cava. And we have a nice picture that does a good job of showing that right here. Notice here, veins are inferior mesenteric vein, superior mesenteric vein, splenic vein, gastric veins. All of them feed into the hepatic portal vein, which feeds into the liver. Magic happens. And then after the magic, it feeds out the vein into the hepatic veins into the inferior vena cava. So don't get these two mixed up. Hepatic portal receives it from the digestive system, feeds it to the liver. Hepatic vein from the liver to the inferior vena cava. A very important specialization to this system to, again, make the blood nutrient appropriate. Add things that are deficient in the food, right? Take things away that are excessive. A vitally important process. All right, that brings us to the legs. The legs, again, are pretty simple and straightforward. A lot of symmetry. And again, just a couple extra blood vessels. Our largest blood vessel in the body happens to be located here in the leg, and it is the great saphenous vein, basically going all the way from the foot all the way up the lateral aspect of the leg until it finally feeds into the femoral vein. This is the one that used to be very commonly harvested for things like those bypass surgeries, because it's a nice long length of vein. Now more and more blood vessels from cadavers are being used for bypasses, but obviously if you're using your own blood vessels, much less of a chance of rejection from that, so that this would commonly be the blood vessel that they would use for those types of processes. And then anytime there's a great something, there is going to be a small something. And so notice on the lateral aspect of the leg there, from basically going from the foot to the popliteal in the knee, is the small saphenous. So we have that small saphenous, anterior tibial, feed into the popliteal, into the femoral, into the external iliac, common iliac, inferior vena cava, back to the heart. So again, nothing really confusing, and complete symmetry between the left and the right for the legs as well. All right, as I mentioned, that is not every blood vessel on your list, but those are most of the idiosyncrasies, the asymmetries, not only between left and right, but also between arteries and veins as well. So we've seen some of those differences. Hopefully those will help you on your pathways, and more importantly, help you on the exam as well. All right, any questions on any of that? So remember that cardiovascular exercise, which is being graded for correctness, is due on Wednesday. If after today's lab, you're concerned about yours, then it's okay to go ahead and start from scratch again, right? Go ahead and work on it, make sure it is correct, because it is going to be graded for correctness. So hopefully those things we talked about today will help. All right. Any other questions? All right, excellent. That is everything that I have for you for today then. So we are done with today's lecture. We're done with today's lab. You are free for the next 2 days. I will see you on Wednesday. All right.
Thank you.
Have a good day. Take care. Talk to you later.